Hello and welcome to Kickback Science Podcast. I'm your host, Dr. Luke. This podcast is your place to get the tools and knowledge necessary to navigate the world of health and wellness. So kick back, let's talk science. This is good content. This stuff has gone crazy. We'll jump into it. Let's dive in. Let's do a little science. I'm going nitty gritty science right now.
I've been looking forward to this.
Kind of going on a little bit of a tangent.
Oh yeah, that's right. I forgot about that.
The information we discuss in this podcast is not meant to be taken as medical advice. It is simply informative. We're not affiliated with any organization or institution, just a few guys talking science and having fun.
All right.
Okay.
Just dive in?
Do it.
Should we just dive in?
Yeah.
What are we talking about today, Luke?
Shut up.
Oh, I'm sorry.
I'm not going to tell you.
All right.
Just don't say shut up to your wives.
Yeah.
Welcome back to another Kickback Science Podcast episode. Another fun one because we got our second guest, a very special guest actually that Reed and I happen to know pretty well. He was my teacher actually in Mankato. Was he ever your teacher?
No, he was my roommate though in South Africa though.
No way. Oh yeah, when we went down to South Africa, did research down there. Well, he's my teacher, so now this is kind of cool because I get to have him on my podcast after learning from him one day. All right, so who the heck is this? Who's this guy on our podcast, right? So this is doctoral candidate Zack Roloff. So Zack was down in Mankato where he got his undergraduate and master's degree in exercise science and then exercise physiology. The exercise physiology, after he got his master's degree, or while he was getting his master's degree, actually, he was teaching exercise physiology labs, and that's when I was a little bitty undergrad student. And he taught me everything I know here today, so he's the reason that we have this episode, I think. Shout out, Zack. And then afterwards, spent some time working. We've talked about VO2 max and those metabolic carts and such. Zack actually worked for a company because this guy could literally build or fix anything that is involved with anything. Like the bod pod for measuring body composition that we had, he could take it apart, put it back together. Used to do that with metabolic carts. And then he decided, you know what? I'm a scientist. So he is now actually getting his just finishing up his PhD at the University of Minnesota. So we're going to hear from Zack a little bit about the research there. All right, Zack, what's up?
Not a lot. One thing you did miss is not only do we know each other very well, but Luke also married me and my wife. So if that tells you, we called him one day after we got engaged. I called Luke, and me and my wife both were like, "Hey, Luke, you want to marry us?" And he did. So that tells you kind of how close we are. That's
that's awesome.
Weddings, baptisms, and funerals. I can do all three. And if you sign up for two, like you guys are about to have a kid, so if you sign up for the podcast, you get the third one free. So I'd do your funeral for free. It's a little deal I'm running for a period of time because we have weddings, baptisms, funerals. So. All right.
The other thing I want to mention too is in circa 2014, must have been 2014 is when I met Reed.
Yep.
Pre-South Africa. And then at the same time, I was teaching Luke in biomechanics and kinesiology. At the time, I was in a master's program and with aspirations to get my PhD. I didn't get into a PhD program, but Luke did. Before I did, I planted the worm in his head to go on and expand your knowledge. Keep going down the route of academia. So Luke gets his PhD. I'm working. Luke finishes his PhD, and then I enter my PhD. It was pretty close to that, but Luke was almost done with his PhD by the time I got into a PhD program. So that's super fun. And that's what we call a segue.
I forgot that was what happened, but when you just said that, it made me think like the reason that I probably got accepted. I mean, PhD is different because, it's dependent on funding, but you were the one who was like, hey, come into the lab. You can help me with my study. You got me a publication and presentation or presenting my research. Yeah. So as you're working on trying to get into a PhD program, I'm piggybacking off everything you're doing. It worked for me. So thanks.
Yeah, first author on this paper, and Luke's a middle author, which is good enough in itself, but Luke gets into his PhD program on being a middle author on my first author paper.
Yeah, so thank you.
And it took me, an extra 4 years to get into a program. But again, that's about funding and whatever. But so what, you want to hear about how I got into a PhD program?
Yep.
It's kind of a cool story.
Yeah, let's hear it.
So I'm in the rehabilitation science program at the University of Minnesota. It is part of the medical school where they have 7 or 9 different PhD programs. And like Luke said, I was working for a company at the time that manufactured cardiorespiratory diagnostic equipment, and one of the labs in the rehab science program had a broken machine. None of the guys wanted to go to the U because this machine in particular was in a basement. The parking sucks, and I was like, yeah, I'll do it. I was working tech support, so I'm on the phones all day long, and any reason to get off the phones I'm like, yep, I'll do it. So I go to the U and find this cart. I'm in there for about 2 hours, and I'm talking to a student that was in the lab at the time, just telling him about kind of what I've done, what I'm doing. And I told him I wanted to get my PhD one day, and he goes, but right before I left, he goes, you want to go meet our program coordinator? So I go upstairs and meet the program coordinator. His name's Rich. He's a great guy. He's been awesome for me over the last 4.5 years. And he sets up an interview with me and the director of graduate studies for rehab science. Her name is Dawn. So I met with Dawn. She sent me off to interview with a couple other PIs in rehab science. One was at the VA. One was in neurophysiology. None of them had funding, but I got an email from Dawn one day. She goes, my grant got renewed and the spot's yours if you want it. So I only ended up, it's like this little bit of serendipity. I got into my PhD program simply because I knew how to fix a metabolic cart is kind of how I draw it up.
Awesome.
So
See,
yeah,
that's like, I mean, a message to anyone who's trying to get into a PhD. I mean, it's different than any other program you're applying to because it's not like they take a certain amount of students per year. You have to luck out. Like Don got funding. If Don didn't have funding, you wouldn't get in no matter how good of an applicant you are. But that's funny. Like, so you're going through all the stress of trying to find a program and then, oh shit, I just got in.
right. And that, that wasn't entirely the route that I had expected to go. It's a physiology lab. There's lots of biochem and muscle physiology stuff that we do, which I'm very comfortable with muscle physiology, but I was not familiar with bench work at all. So I thought I was going to end up doing like exercise studies with animals or bench work with humans. I had already worked with people before.
What's bench work for the listeners?
Bench work is like doing assays and working with more kind of on a cellular or tissue specific kind of what you see in, if you see like a stock photo of people in a lab.
Yeah, that's exactly what I was going to say.
That's bench work, where they're looking at little test tubes and using the centrifuges and cell culture hoods and all of these things. So I thought I had done human performance research with high level athletes. So I could work with humans already, and I could work with the VO2 maxes and all of that. I wanted to lose one of those elements. I wanted to lose the human part and work with animals and still doing training studies, or lose the training studies, keep the humans, but doing more bench work. Instead, I got animals and bench work, and I had to learn on the fly. And of course, I joined my program in January of 2020, which was not a great time to start a PhD program.
That's tough.
And like I said, if I had been able to work with humans and done the bench work or animals and I'd only have to learn half, right? I could focus my work on learning how to do these different biochem assays or whatever. But I had to learn both, and I was in the lab the beginning of January, and then I think I was out the first week of March. So all I had learned how to do was read the instructions on how to genotype some mice. Like that is the extent to the lab work that I had up to that point. So
Damn.
yeah, it was a rough time, but looking back and knowing what I know now, you can look back and you're like, well, the first 2.5 years of your PhD program is all taking classes.
Yeah.
Typically you're doing your lab stuff on top of taking those classes, but instead I got to really focus on my lab or on my classes. So that was nice. I got to really engage in those classes and learn a lot. And I took a microscopy course that certainly would have been better suited to being in person, but I took it online, and I learned the inner workings of fluorescent microscopy and using confocal microscopes. And that kind of leads into where my research was headed, was because I am interested in devices already, and using a microscope of that caliber, of that extent, was very interesting to me. That's another segue. But
was that in your, yeah? So was that in the second year, and then
no,
segued into you kind of following this path towards your dissertation?
So I'm trying to remember. I think my first year was I was there for classes for the first, whatever it was, 6 to 8 weeks, and I think that summer was when I took that microscopy course, because the following year I started going into the lab a little bit more, but I know I took the microscopy course over the summer. So it really, actually thinking about it, yeah, I had my first semester mostly in person. The second half-ish was online. I took that microscopy course over the summer, and then in that fall, I took our grant writing course, and that grant writing course snowballed. Like, I had some ideas. I didn't really know a lot about what I was going to be studying, but I was taking over a project from a previous student, and the grant writing course really made me think about what questions do I want to answer. So a lot of the stuff that I ended up actually doing was the foundations of that were set in that grant writing course in September of 2020.
And so then we'll segue into like, well, what the hell are you doing now?
Yeah.
Like, what did it lead you into doing the research? But for people, we've talked about this in another episode, but a PhD is super unique in terms of like, yeah, you have the couple years of coursework, but you have to come up with and conduct a study. So Zach's saying he's, coming up with asking a question. Well, that's your job as a PhD student, is to like figure out what question do you want to ask that hasn't been asked before that you're interested in. And then you got to design an experiment to answer that question. So you have to conduct this research. And that's what your dissertation is. So hit us with the dissertation, Zach. What the heck are you doing?
What am I doing?
Yeah.
Okay.
Or however you want to take that question.
Yeah. So I work in a muscle physiology lab that is generally been looking at in vivo or in vitro muscle physiology. So in vivo is, the way I think of it is, they're live animals. They're in vivo. Vivo is life, like in Spanish, vivir, to live. So in vivo. So I think that's how I remember the difference between in vivo and in vitro. So in vivo, the animals are still alive. They're anesthetized, but we use these probes to, we put them into the muscle, and we can do muscle contractions. So they're looking at isometric force and twitch force, and they do eccentric contractions, so lengthening muscle contractions to look at muscle damage. In vitro, we can do the same thing, but instead of it being a live animal, we have live muscle. So we euthanize the animal, and we'll take out whatever muscle the people are interested in. So generally it's the EDL muscle, it's the extensor digitorum longus, which is on the mice, it's a hind limb muscle. They will tie that into this bath of a buffer that oxygenates the muscle, and they can do the same exact thing that they would with an in vivo model with just the single muscle, right? Same thing, lengthening contractions, things like that. So that's kind of the majority of the research that's done in there is that kind of muscle physiology stuff. We also do some aging research, and we use an overactomized model of mice where we do a bilateral removal of the ovaries in female mice to study estrogen deficiency and estrogen loss. And then for me, you get all these parameters that you're looking at with these groups, with the other researchers in my lab. They're looking at estrogen deficiency and isometric force and all these. When I explain my research, you'll understand why I don't really know what other people are doing in the lab. So they kind of have this bubble where they all know what each other is doing. It's not a very big lab, but I work with muscle that is relaxed, non-contracting, and in relaxed muscle, I take apart these muscles, and I zoom in very closely on them under this super fancy microscope. What I do is look at relaxed myosin. Myosin is a molecular motor in muscle that causes muscle to contract and shorten. When muscle is contracting, you are turning over ATP, which is the unit of energy in muscle. Generally, we think of ATP utilization in contracting muscle. However, when muscle is relaxed, they're still turning over ATP. So this is ATP hydrolysis, breaking down ATP. And in relaxed muscle, it exists in two distinct states that they're characterized by the shape of the myosin. So if you think of myosin kind of shaped like a tadpole, kind of there's a head and then it's got a tail. That's all there is to it. When the myosin head is straight up and down, it is in what is called the disordered relaxed state, and it's turning over ATP slowly and much more slowly than it would be in actively contracting muscle.
So it's using energy at this point. It's just not to the extent that energy would be used
Correct.
if you were like working out or
Correct.
moving the
Exactly, yeah. So in actively contracting muscle, one ATP molecule is being hydrolyzed less than a second. We're talking a fraction of a second ATP utilization happens. In the disordered relaxed state, it can take as long as 60 seconds for one ATP molecule to turn over, depending on the animal or what we found it's in humans as well. The other distinct relaxed state that we see is called the super relaxed state. And what happens is that if you think of that tadpole again, think of a tadpole kind of folding in half, where the head is going to touch the tail, right there. So these myosin heads will fold back in on themselves. And what that creates is an inhibition of ATP turnover. It slows down the utilization of energy. I think of that as a product of the need for the conservation of energy. So you think about skeletal muscle, it takes about 40% of your total body mass. So
Is that true? Wow.
Yeah, it's about 40%. Huge organ, right? And it's got crazy energy demands. It's everything that you're holding a cup of water, you're using ATP right now. You're sitting doing nothing, Reed, and you're using ATP right now.
Yes. Reed's always fidgeting though.
Yeah.
So.
We might get into that later.
Yeah.
So because of the way that muscle utilizes energy and the overall volume of muscle that you have in your body, it has this massive demand for energy. However, even though the volume of muscle in your body is large, the rate at which the basal metabolic rate is low comparatively to the volume of the muscle, right? So they think that happens because of this super relaxed state. So if the majority of myosins at rest, at in vivo temperatures, so our in vivo temperature is 37 degrees Celsius, something like that. Because of the low basal metabolic rate of muscle, they think that the majority of myosins are in the super relaxed state because ATP turnover happens 200 seconds or more. One ATP molecule is being turned over at about 200 seconds, 230. I've seen as long as 1,000 in certain animals. And being that ATP turnover is slower, muscle thermogenesis is also lower. So muscle thermogenesis being the heat production of muscle. If ATP is not being turned over as quickly, muscle thermogenesis is going down or staying low, then also your basal metabolic rate, that resting metabolic rate, will be low. There's kind of this theoretical cascade of events where
Makes sense.
Myosin is folding back on itself, ATP turnover is inhibited, muscle thermogenesis remains low, and downstream whole body metabolism is low as well.
So if you think of this from like a mechanistic standpoint, like why the hell would we have this like state of the tadpole fold. It made me think of Cato when he takes a nap. You know, he's kind of like folding in on himself. He's sleeping, right? He's resting. So, like, why the hell would our body need this? You're saying it's from an energy conservation standpoint, which, like, evolutionarily, we, you know, didn't have access to all the food we do now, and so that makes sense. I had a question I was going to go to, but now I'm not. You keep going. I'll think of it.
So, just getting into basal metabolic rate a little bit, right? So, basal metabolic rate is the rate, your rate of metabolism of just your normal involuntary bodily functions, right? You have smooth muscle that you have no control of that's turning over ATP. That counts towards your basal metabolic rate. Your breathing, you know, all of your organs in there are using ATP, and you have this generally low rate of metabolism just at rest. And that we think that is somehow related to the kinetic states of myosin. We don't know exactly how they're related. So that's one thing I should mention too, is that the history of the super relaxed state research goes back only to 2010, right? So this is a relatively new area of research. The researchers that discovered this are out of San Francisco. They noticed that when they had purified myosin in culture, there was a ATP turnover similar to what you see in the disordered relaxed state. So purified myosin turning over ATP roughly 6 seconds is what they found, 6 to 15 seconds, something like that. But when they actually used frog muscle to measure ATP turnover, there was this vast difference between the purified myosin and the frog muscle. So they developed this experiment to measure the rate of ATP hydrolysis in muscle. So what they do is they will take a muscle bundle. I use mice, so I'm going to talk about the mice that I use. So I'll cut out the soleus muscle. The soleus is behind, it's one of your two calf muscles. It's behind deep to the gastrocnemius muscle. So if you look at your calf, you have these two heads, and then tucked deep under that is this muscle that is mostly used for kind of stability, and it's not really a muscle that you think of going to the gym. I'm gonna get my soleus looking great. Right? It's not one of those. I use this muscle because of its. In a mouse, it has varied fiber types, right? We know that there are different fiber types. Some are some utilize energy, carbohydrates more as energy. Some will utilize fats better for their energy, that kind of thing. So type 1, type 2 muscle fibers. The soleus muscle has a combination of type 1 and type 2 fibers, which is why I used it. I'll take that muscle and I will permeabilize it. So I put it in a solution to remove all of the membranes. It removes connective tissue, it removes all of this stuff. So all that I'm left with are essentially the contractile proteins of the muscle. Specifically, what I'm looking at are actin and myosin, and really mostly just myosin. So once that muscle is permeabilized, I put it under a microscope and I can isolate single muscle fibers out of that. So one muscle fiber is a muscle cell, and they're very, very tiny.
Yeah.
So you pulled them out. Actually, you did that, right?
Yeah.
I had you do that before you started at the
Yeah, I saw this crazy. They have like a room just dedicated to this one giant microscope for his research. Yeah. It's wild.
So I'll isolate these fibers, and then I put them on a microscope like cover slip. And then I have to isolate all my fibers, and then I bring them down from my lab to another room where they have this fluorescent microscope that has the exact mechanisms that I need to measure what I want to measure. And what I end up doing is putting this fiber on a microscope, and I incubate the fiber in what's called MANT ATP. It's got all of the same stuff as normal ATP, except for the MANT part of it increases the quantum yield, which increases the quantum yield of the ATP, meaning that it will fluoresce under a microscope.
So you can see it.
Yeah, so we all have, everything's got a quantum yield to some extent, right? Everything will, to some extent, fluoresce under a microscope. ATP, however, is not a very high quantum yield, so we can't see it very well. So they have found this analog that allows us to see it bind to muscle brightly under a microscope. And so I will incubate the fiber in MANT ATP. I'll remove it and exchange it with an unlabeled ATP, just normal ATP. And I measure the decay. Because the normal ATP is what the muscle is expecting, the binding affinity to myosin is much higher in that unlabeled ATP relative to the MANT ATP. So because there's competition for the binding sites, the unlabeled ATP is going to win, and we get a fluorescent decay over the course of 8 minutes. And from those 8 minutes, I take that data and I plug them into a software that gives me the different variables that I'm looking for. So the decay is a 3 exponential decay function. This sounds very, very high. Like, I'm not this smart, I promise you.
He is.
There's a software that does all of this for me. And the way that this formula is set up, it's got amplitudes and it's got rates. So the rates are the rates at which one ATP molecule is turned over, and the amplitudes are the ratio of myosin in each respective state. So I get a ratio, I get a proportion of myosin in the DRX, the disordered relaxed state, and I get a proportion of myosin that are in the SRX, the super relaxed state. And those values together equal 1. And then those rates, I take the inverse of the rates to get the lifetime. So those lifetimes give me the amount of time it takes really to go through that ATPase cycle. And this is a lot, but I have a lot of tools that do the work for me. So what I end up with, this is kind of what I'm getting at here, what I end up with is I get a fraction of myosin in both the DRX and the SRX. Those are my primary variables of interest are these fractions. But then I get the lifetimes as well, where I was talking about ATP turnover happening every 15 seconds in myosin in the DRX. That's what that lifetime is, that 15, 30 seconds, 70, 75 seconds, whatever it is. And then the SRX, I can get up as high as, I've seen as high as like 450 seconds, which is the entire length of my chase. I go 7.5 minutes. If it goes over that, I wasn't really counting.
And is this for one ATP, you're saying?
No. So
that 450 seconds.
No, that 450 seconds is the entire chase of it's like we call it a chase because the unlabeled ATP is chasing away the MANT ATP out of the binding sites, right?
Gotcha. Okay, so that's what you were talking about the time period. But you said like the difference between actual ATP hydrolysis in the disordered versus super relaxed state. Like, did you say like 60 seconds sometimes in the super relaxed state for one
molecule? Yeah.
So when you said 450, I thought, but you're talking about the chase experiment specifically.
Yeah. But in the super relaxed state, I have some data that show that these specific myosins were turning over one ATP molecule in 430 seconds.
Okay, that's wild.
It's wild, yeah. The inhibition of the ATP turnover by the way that the myosin is shaped is incredible. And if you think about if you can conceptualize the numbers of myosin, even in DRX, those numbers relative to what you see in actively contracting muscle, it's clear to see why that this is pertinent to studying muscle and muscle metabolism, right? Where if we can harness that, if we can harness the super relaxed state, if we can target that with a pharmaceutical and we can shift myosin out of one state to another, specifically, and what I want to see is being able to shift myosin out of the SRX to the DRX. Well, if you can go from 400 seconds to 30, we're going to be increasing ATP turnover by a lot.
Basically using a drug just to ramp up metabolic rate.
Exactly.
Yeah.
Exactly.
Okay.
Yeah.
That's wild. So your experiment specifically, like you're isolating now, what groups are you comparing here?
That's a good question. So my first study, I looked at muscle from mice and rats. And I did this spiel about how I wanted to use mice because I wanted to use their soleus, blah, blah, blah. I used rats for the exact opposite reason, because their soleus muscle only has type 1 fibers. And in general, you're looking at, again, to conceptualize this for human muscle, if you take someone who looks like me, I do
Jacked.
Sexiest soleus in the game.
Thanks, Reed.
Killer calves.
Somebody who is more built for resistance training and short bouts of high intensity exercise versus somebody who, even like Luke, can just run.
Not anymore. Those are the old days.
Never mind. So if you have somebody who has these the differences in physique or stature can be kind of a way to look at muscle fiber types, right? I'm generally, I would imagine if we were to do a muscle biopsy from me and a long distance runner, you're going to get type 2 fibers in my muscle, type 1 fibers in the long distance runner, just the way that they're utilizing energy. The reason I use the rats is because their soleus muscle, that's been found that they are virtually almost all type 1 fibers. So you can eliminate some variability by using a rat soleus muscle because you're only dealing with one fiber type. So one of our collaborators they do all mice work. It's actually the guy that I interviewed with who does neurophys. Being that they do neuro work, they don't need the muscle. They were doing this study. They called me and I was like, yeah, I'll take some muscle. They had fed these rats what they called a Western-like diet. It's relatively high in fat.
McDonald's,
right? Sugar, yes. Lots of sugar.
McDonald's, hamburger helper. We're in the Midwest, so you get some tater tot hot dish mix in there. These rats are living the dream. So they get, I think, if I remember right, that diet was 38.5% from fat. Right? That might be too high. Doesn't matter. Relatively high in fat.
How do they feed them? Do they actually chop up burgers and or what do they? How do they feed them?
So
puree stuff and just pour it.
The rats, I don't have as much experience with the rats, but with the mice, they have these pellets that look like essentially they look like dog food, but just kind of in a longer log shape. And there are companies out there that will customize these pellets to have the exact composition that you're looking for.
Oh, wow.
So cool. The rats, I would imagine that they eat pellets too.
They do.
I didn't see them until they were dead. So
we used to do rats out in
Okay.
Colorado. Yeah, same thing. You just, that'd be cool though, to give them actual Big Macs.
Yeah,
but essentially they're eating the same thing. They think it's the same thing composition wise.
Yeah.
Yeah. So if you can
it's
look,
actually different.
Yeah. So the companies will send you the composition of the diets that you're looking at. So the rat one, you look at it, it's 6 ingredients, and it just says protein and lard and cellulose and vitamin mix, and it's that's all there is to it. So that's how they get their fat is from lard. So these rats, I had male and female rats that were fed this Western diet for 12 weeks. So when they were euthanized, they were roughly equivalent to a 20 or 22-year-old human. So prime of their lives. And we took out the soleus muscle, went through that whole process, permeabilized them. And then we did the same thing with mice. Another collaborator had mice that were on a high-fat diet. They were working with HFpEF. They're studying HFpEF in this lab. So they're heart failure, preserved ejection fraction. Yeah. They're studying metabolic syndrome and how that's related to HFpEF. But they too don't need the muscle. They're looking at the heart. They're doing brain stuff. They called and asked if I wanted to take muscle, and so we went out there. And these mice were on a high-fat diet that was. Trying to remember the numbers here. I want to say it was almost 50% from fat. That diet was, and they were on it for over 20 weeks.
Wow!
So these little mice that generally are 35, 40 grams of that's normal body weight at their peak, 40-ish grams. I had one that was pushing 60 grams.
Wow.
Yeah. Big boy.
So you got a bunch of dieting experiments that you're studying.
Yeah. Okay. And I have a hard time I have a hard time calling it a diet study. When you start talking about diets, you start talking about obesity, and there's so many steps in between what I'm doing and actual obesity that I have a hard time calling what I have done obesity studies. For the layman, it makes sense to call it that because it's just easier than what I had to write about in this paper was I talked about fat-inducing condition, because I'm looking at the effects of increased adiposity at the muscular level and how that changes the parameters of relaxed myosin.
Gotcha.
But
okay.
In a sense, it's an obesity study, right?
When you zoom out,
yeah,
you're just a little deep in the mechanisms.
Yeah,
behind.
and part of that too is probably naivety on my part. Again, not really knowing what I'm doing. There are, similar to BMI in humans, there are scales that you can use in rodents that you have to measure from their nose to their anus, and that gives you what that length, and their weight gives you some kind of body mass index. I didn't do that. I didn't know that existed, and by the time I realized that was a thing, I didn't have the animals anymore. So we feel that we can't really call this an obesity study. We can relate it to obesity, and there's some bridging that has to happen there, but for all intents and purposes, that's really what we're looking at. Just scientifically, journalistically, it's a little more difficult to do that. But so I had male and female rats, male and female mice, and you go through all of these fibers. Now, keep in mind, so I have, what is this? I had a 12, 10, I had a lot of animals. That's what I'm getting. A lot of animals. I took out the soleus muscle from each one of those animals. I actually took out the gastrocnemius from each one too, just because I'm already there.
Did you do the EDL?
I did not do the EDL. Mostly.
We used to do that, like, for whatever reason, any experiment we did with animals in Colorado, it was just like standard. You take out the soleus, EDL, gastroc, you toss it in a freezer, and then someone's going to use it down the line.
Right. Yeah, and I probably should have done that. We didn't know. There's not very many people that are doing this kind of research, so that some of these things are just like
taking up space in the freezer. Then.
Well, not even that, but just because there's not a lot of this going on, you just don't know what's important to take. Specifically in the world of relaxed myosins, but. I mean, looking back, we probably should have just shredded these animals and taken as much tissue as possible, muscle tissue specifically as possible. So yeah, we had I had 4 groups per study. Both of these studies are going into one paper. And so I'm doing male-female comparisons, and for the sake of what we're talking about, a high-fat and low-fat diet comparison. And so we're running 2x2 ANOVAs to analyze these data, and it's a lot of data. There were 20-ish animals. I took this. My main muscle of interest was the soleus, and I took 8 to 10 fibers per muscle. Each muscle takes about 15 minutes to get my data.
That's a long project.
Oh,
and a lot of data.
Not to mention the microscope that I used costs $30 to $50 an hour.
Oh, jeez. Oh, and then so you gotta have the grant fund. Well, good thing you're in an awesome lab with a lot of money.
So, shout out to Don Lau, Dr. Don Lau. Very well funded, and has been great to me. I feel like the longer I'm in the lab, I realize how lucky I really am to be in there. So it's been very nice to be in a lab that has been well funded. And you need some microscope time. Go ahead. It's $30 an hour. Go do your thing. So I would schedule for about 4 hours, 6 hours at a time, and get 3 fibers done in an hour. It's a long process.
Yeah.
So you get 3 fibers done in an hour, but that doesn't mean that they're going to work, right? I could go through the whole process and look like my data are good, but then I go and actually run fit the data to an exponential function, and that part doesn't work.
Dude, that's like, I contemplated dropping out of my PhD because of that exact thing, where you go through this whole process, and there's a lot of experiments and procedures that lead up to your final analysis or imaging or whatever you're doing. And I was doing PCR, and we were doing like a technique that we hadn't done in that lab, and it was my job to figure out if it would even work. It would just never work. You go through days of this process of collecting your data and then doing all these tests, and then finally the imaging, you just cross your fingers, and it just wasn't working. Finally one day it was like, boom, it worked. And then it was, I got to figure out, why did that work that time? Oh man.
So you were staining muscle or staining tissue to image? Like, were you doing like histology?
I was actually using blood samples.
Oh,
so
okay.
yeah, we were looking at gene expression, the circadian rhythm gene expression, circulating around in blood. But yeah, just the way that we were doing it was a different technique. Yeah. So it was funding, dude. Like, if we had the funding, we would have been able to do these full blood draws, and this process would have been easy. But I'm in a lab that, you know, like, I had to try to scrape together all these grants, and we couldn't afford to do that. Right. So we had to do figure out how to get to the end. And so dude, you tell me this gives me some PTSD.
Were you doing humans at this point, or was this on the rat?
Yep, this was humans.
Okay.
Yeah, I couldn't do the animals anymore.
So the thing about the rats that I learned was awesome is, you know, you have these pros and cons of using different animals, right? Like a rat is much bigger than a mouse. And if you have a muscle that you're working with, you can use that muscle for a long time. There's a lot of stuff there that you can use. Mm-hmm. A mouse, you can't do that. With the rats, I could take one soleus, cut it longitudinally, and have essentially 2 soleus muscles from one side of one rat to do fiber, like my fiber experiments, plus an assay or something. You know, with a mouse,
if you mess up,
there
goes that mouse. Right. And then that's what I felt so bad about.
Well,
too, it's like, that's a waste of a mouse.
the rats, you know, the rat tissue is great, but I don't think I could take the rat and euthanize it. I think I couldn't handle working with the rats.
Because someone else is doing that.
Everyone else, yeah. So this study, everyone else is doing all the other stuff. I didn't have to deal with the diet. I didn't have to deal with tending to the animals, anything like that. I just took the muscle after the animals were dead. I don't think I could euthanize a rat.
It's not fun.
They look like they're so much fun. I just want to play with those because they are. They're so they were Sprague-Dawley rats. So you think of a white lab rat. That's what they're using. They're so friendly. My friend who was working with me on this study, she was the one in the other lab doing the actual rat work. She would.
And there goes the mic.
Welcome to podcasting, Luke.
All right, we're good.
So Rebecca is a friend of mine who is now at University of Michigan doing a postdoc, and she would call these rats, she'd be like, "Oh, come here, baby." You can't talk to the rats like this. They're gonna die in 40 seconds. Stop it?
Come here, baby.
Yeah, that was so. Shout out Leah. Leah was another PhD student in my lab, and she did the same thing. Hold it, and she said she had a pet rat before too.
Yeah, how could you do that?
I'm like, you just shouldn't be working with rats.
No way. And so the thing about the mice then is the opposite. You have very limited tissue, and good luck to you. But mice are dicks. I don't care about. I don't care about. Shouldn't say that. I do care.
Shout out PETA.
We're actually sponsored by them. Yeah.
So. PETA kills more animals than anybody else on the planet.
Ooh, hot take.
It doesn't affect me the same way that I think a rat would. Maybe a rat. I don't know. I didn't have to deal with them, but it doesn't. I don't have that emotional.
I know what you mean.
I almost cussed.
You can cuss. You can fucking cuss.
Those fuckers will bite you, and it hurts. And I'm lucky enough to not have to deal with the mice a ton, so I've not been bit a lot, but they'll bite you, and it hurts. And they're dicks.
Yeah.
So you have these give and takes with these different animals. What was I talking about before this?
Dude, I was talking before to Reed about how you and I are so similar in terms of our brains are so disorganized, because we were laughing about how last night you were like, wait, I'm coming over. Well, I guess it wasn't that I didn't think I was. I just didn't know. I didn't know. So these what you just did, every single episode, that's what I do. I just start going and going.
I told Emily, I go, this is the text that I sent Luke after he sent me his address was, that's so like me. Us, actually. Yeah. It's us. This is us.
Yeah.
It is the microcosm of who I am as a person. And doing a little callback here, when Luke did our wedding ceremony, he made sure to put that in there.
Yeah.
The difference between me and my wife is that Emily would have been well prepared.
Yeah. Perfect script
notes.
But this is how we do it on Kickback Science. We just go.
So
Let me bring you back to like where I see this going. So what I want to do is come back to, first off, like, what was your hypothesis? So I think we understand the experiment at this point. Tell us your hypothesis. And then after we talk about the hypothesis, I want you to give me like a minute of everything we just covered. How could you say that to someone or summarize it and why it matters?
Sure.
Why are you doing this experiment? Why do we care about the myosin state? And then we'll segue into, kind of coming up back to surface level.
Sure.
So first tell us your hypothesis and then let's summarize.
Sure. So with this particular study, with the adiposity study, part of the problem with this, here's the preface, is we have 4 variables that we're looking at. 2 SRX variables, 2 DRX variables. And I think about this in terms of the stability of the super relaxed state. I care mostly about that state because of the inhibition of ATP. So if we destabilize the SRX, that means the myosin heads are unfolding and they're shifting to the DRX. Or the SRX lifetime is shortening. So the rate at which ATP is being turned over is happening more quickly if we destabilize the SRX, right? So
if you destabilize the SRX, are you entering a disordered relaxed state? Like, is there like a threshold at which or is it actually the like mechanical position of that myosin head that I did? Okay.
Yes. So it is the if we're talking about the fractions of myosin, if the myosin heads are being destabilized out of the SRX, the only direction that they can go is in the DRX.
Sure.
SRX to DRX. And the thing that I'm running into now is how important is the lifetime? And I think I'm learning now that it's more important than I initially expected. When it gets convoluted, it's tricky because you have 4 variables that are working tangentially in tandem, but also kind of tangentially because they can go in opposite directions. It's difficult to quantify and think about, okay, so this number went this way, but the other one went the other way. So what does that mean as far as total ATP turnover? It may mean nothing. If SRX myosin shifts out towards the DRX but the SRX lifetime gets increased, that there's still a low rate of ATP turnover there, right? I'm kind of, that's where I'm at right now, is figuring that out. But
could you quantify, like, in the end, the total amount of like estimated ATP turnover? And then wouldn't that really be the end result of what you'd want to look at anyways?
100%. Yeah. So there have been studies that have estimated that number, total ATP turnover, but you have to make assumptions when you do that. And
so you got to be the guy to come up with some sort of algorithm.
Yeah. So I don't know. Have you heard the name Julian Okala?
Nope.
Do you know who that is?
Nope.
So the Okala lab is in Copenhagen, I think, and they're working on skeletal muscle SRX studies, and they have made those assumptions for us. But I don't know enough about I don't know how to put it. They assume that there is a certain concentration of myosin in muscle, and that like there's 220 micromolar myosin in this particular myofibrillar thing that they have. Like, they're just studying myofibrils. So they make this number 220 micromolar. I don't know if I can do that with mine. I doubt I can. But that's the trick, is if I can get this concentration, I can relate that to my lifetimes and my rate or my fractions of myosin and get total ATP turnover.
Sure.
But to answer your question,
that's just another variable. Like, someone else will do that. You don't.
Right. To answer your question, I initially hypothesized that the SRX would be destabilized with as a result of the high-fat diet. Increased adiposity in these animals, I thought would shift myosin out of the SRX to the DRX. And I can see the face that you're making, and I make that face regularly because
it seems counterintuitive.
Yeah, right. But when I first started doing this, I thought of the SRX as kind of an efficiency mechanism. Well, when you are inducing adiposity, when you're inducing obesity in these animals, your muscle is going to be less efficient, right? Like in an obese person, muscle is less efficient. And because of that, I thought the efficiency was the SRX being lost. What I'm finding now is that that's not necessarily the case, is that maybe the SRX is an efficiency mechanism, but only on top of other things that matter more, right? So my hypothesis was that the DRX would be, or the SRX would be destabilized as a result of increased adiposity in mice and rats.
So that to me, I mean, you explaining it does make sense. And like to have efficiency in any physiological function in the body, you know, as you become obese, everything's going to become impaired. Is that why we see, I don't want to get at this quite yet. I'm gonna follow up in a minute about like obesity and metabolic rates.
And we call that a
but
tease in the business.
Yeah.
We'll circle back.
We'll circle on back. That's I always say that. You gotta circle me back.
Does Reed takes the notes of the circles?
Yeah, because sometimes we don't circle back to them.
I just come up with something like that, like it pops into my head, and I say it, and I'm like, "Ooh, that's better for later." All right. Anyways,
so what else? So you want kind of a 3-minute thesis-esque. Why does this matter?
For someone who has never taken a science class.
Sure. Okay. So, funny enough, I did a 3-minute thesis.
Didn't you win that?
I won it.
Yeah.
So, we didn't, because of the way that I talk about my work, my brain is so scattered. I didn't really get into thermogenesis, and I think that's an important mechanism that's involved here. We talked about it, I brought it up, but what's important to know is that there are different ways that muscle can produce heat. And there is diet-induced thermogenesis. There's a postprandial increase in thermogenesis. So you eat, and acutely you increase your heat production. Mm-hmm. There is exercise-induced thermogenesis, and I think that one is pretty self-explanatory.
Yeah.
But then there's also what they call NEAT, non-exercise activity thermogenesis. And that is everything else. All of your body processes that you're doing, your normal, I'm going to use this phrase, activities of daily living. That's not probably not the right context for that, but that's really what it is. So we're just sitting here. There is some thermogenesis happening because we're utilizing ATP. What happens with ATP is when it's hydrolyzed, you break a bond. And when that bond is broken, it produces heat. And so for somebody who is not in the world of science in general, and specifically not in muscle physiology, if you think about muscle as a motorcycle.
I love where this is going.
Yeah, so keep going, baby.
So think about muscle as a motorcycle. Actively contracting muscle is burning through ATP incredibly quickly. It needs to, because of the energy demands of whatever you're doing. That is you flying down the freeway on a motorcycle, right? Pretty easy analogy. Going
burning
fast,
gas.
Yeah. So burning through that ATP super quickly, burning through fuel, going fast on a motorcycle. When you're thinking about a relaxed muscle, the disordered relaxed state is like that motorcycle at a red light in idle, utilizing fuel, but not nearly as quickly as you would be if you're flying down the freeway. Now, a super relaxed state is that motorcycle is parked in the garage. Off. Kind of.
That's neat.
So the super relaxed state is still utilizing energy. Myosin in the super relaxed state is still turning over ATP, but it's so incredibly slow that it is the rate at which thermogenesis happens with myosin in the super relaxed state is pretty low.
Negligible.
Yeah.
So we could, for analogy's sake, say it's like parked in the garage off.
Right.
Knowing that, sure, there might still.
Yeah, right.
Yeah.
So for the analogy, actively contracting muscle is very fast motorcycle. The disordered relaxed is in idle, and the super relaxed state is motorcycle off, parked in the garage.
That's a phenomenal.
Nice
I was
job.
just thinking.
How can we
I
tie that
should
to
have
Game of
asked.
Thrones?
I was just gonna say.
How can we
So,
relate that to Game of Thrones?
There was one episode I did a Game of Thrones analogy, and then like the following episode I explained. So I was like, you know what, Game of Thrones. I was just thinking as you did. I should have asked you, Game of Thrones. Watch him think one.
I like us mixing it up though, because otherwise people that don't watch Game of Thrones are like, "What are they talking about?"
Yeah,
Wall?
either Game of Thrones or
White
motorcycles.
Walkers?
Yeah, yeah. If you don't know those two analogies,
You're out of luck.
Yeah,
Don't listen to our podcast.
I'm trying to think of
got it. Well, I got an idea of it. So White Walkers, dude, they're dormant for so long.
Right,
I mean,
right,
that's SRX right
right
there.
Right.
Disordered.
I'm thinking of Arya doing her training with the
Oh yeah.
what is his name?
That was
The
a
Faceless
ma-
Man?
The
The
Faceless Man. Yeah. Because it's all about efficient movement, slowly, just still moving but slowly,
Arya Stark.
and then just active fighting is active contracting
I'm
muscle.
actually on the episode right now.
Yeah.
She's training.
She-
She's perfect.
She's
Now
the
you're gonna be
best.
thinking about myosin.
Yeah.
isn't it?
She's disordered. So yeah,
I totally cut you off. Sorry.
No, Arya.
I love that analogy. though.
Arya is the best. Yeah. So the motorcycle thing is what I used for my 3-minute thesis, which is what
okay.
what that's intended for, is getting an audience to buy into what you're saying, to talk about that at a layman's level, right? So you think about the motorcycle analogy. We care about the states of myosin because of the changes in thermogenesis that can happen. And as thermogenesis changes, whole body metabolism will also change. And what we're most interested in is what I'm most interested, I guess, is that if we can target super relaxed myosin with a pharmaceutical, with some kind of therapy, if we can target those myosin and shift out of the SRX to the DRX, we can increase thermogenesis, thereby increasing whole body metabolism. And in conjunction with a good diet, regular exercise, that may lead to improvements in metabolic diseases like obesity, type 2 diabetes, insulin resistance, any of those.
Oh, wow.
Any of those things that you see that are combined to make metabolic syndrome.
Yeah.
If we can target SRX to shift out into the DRX, somewhere down the road, I think we can start treating metabolic diseases at the muscle level in conjunction with good diet, regular exercise. Mm-hmm. I don't think we can do this alone. With
There's not going to be a pill that's just going to do, but
Right.
like that will be the component. And we've talked on this podcast a bunch about like any of those chronic conditions, like you're talking about, metabolically related, obesity, type 2 diabetes, like spiral into this like funnel of every other disease. And it all stems back from this excess calorie, this storage state. And so if you're ramping up caloric expenditure in addition to exercise and still not overeating like people tend to do, I mean, that's huge. But then that like kind of brings me to how much of your metabolic rate is myosin, this disordered rash, like,
Yeah.
you know what I mean?
Yeah. So getting down to the molecular level of myosin, it's hard to quantify. Again,
Ballpark.
But if we talk about muscle specifically, I did some easy math because I knew this was coming. People are so dramatically different in their body compositions that there's a reason why we don't have this generalized number of resting or metabolic rate specifically from muscle.
Sure.
We have a 40% of total body mass is from muscle. We have 2,000 calories as your general, the daily caloric intake, whatever, is 2,000 calories, whatever. But what I've figured out is that resting metabolic rate accounts for about 60 to 75% of your energy, your total daily energy expenditure. And of that, muscle is responsible for 20 to 25%.
Wow.
So we talk just simple numbers. Take 2,000 calories, the recommended daily caloric intake, whatever, right? That's how much energy you burn in a day, 2,000 calories. You take 75% of that, that's 1,500 calories that's just from resting energy expenditure, right?
Yep.
If you take 25% of 1,500, that's 375 calories. That comes from muscle, the energy expenditure from muscle, 375 calories. And that is roughly 20-ish% of your total daily energy expenditure, 18, 19%, whatever. So at the myosin level, I don't know, but muscle in general, your total daily energy expenditure it's about 20% of that. Yeah, it is significant. And again, that comes from the volume of muscle in your body and just the demands of the organ. The brain is is an organ that demands a ton of energy. Mm-hmm. It requires, there's a huge part of that comes, what's left over, it comes from just the brain alone. Mm-hmm. But relative to muscle volume, right, the brain is tiny. And so the muscle takes up a large portion of your total daily energy expenditure.
So you said muscle, 20% of daily energy expenditure. Now, how can that, if you were to induce this disordered relaxed state, take super relaxed state and help aid in this highly metabolic rate that myosin induces, mm-hmm. how much could you change that number specifically?
Sure.
If you're going to target that pharmaceutically.
That's a great question. And they have, so Roger Cook is the guy who discovered the super relaxed state, gave it a name in 2010. His lab, they've done incredible work. They're a bunch of biophysicists. They're so smart. He did some theoretical math with the heat production from one ATP molecule being hydrolyzed. So he's got all of this math that he's done. And so theoretically, I don't know if this has actually been measured, and we can get into that later about some studies that I actually would love to do if I had the funding. But so say you take, let's see if I remember these numbers right. I think it was 20% of the myosin that is in SRX, and you shift it to DRX. You're increasing the rate of muscle thermogenesis. I think it's double. You double the rate of thermogenesis in muscle if you shift only 20% of myosin.
Jeez.
Now, you extrapolate that to 100% of myosin shifting out of SRX to DRX. All myosin that is not actively bound to actin. If all of it is in DRX, you're going to increase muscle thermogenesis by 80%.
That's insane.
And increasing whole body metabolism. I'm not going to remember. No. Muscle thermogenesis will increase 4 to 5 fold, increasing whole body metabolism by 80%.
What?
Yes.
Dude, that's wild.
And this is all theoretical. It's not been measured before.
Sure, but I mean, like that, you've got these estimates of like how big a role that this plays.
Right. So,
that's crazy.
And physiologically speaking, I don't think it's probably good for myosin to be. Absolutes are never good, right?
Yeah.
So
there's a reason that this process exists, right?
So, but yeah, that goes to show you the theoretical importance of knowing the relaxed states of myosin. If we can target that, there are some serious shifts that can happen and serious implications to metabolism if we can target this. And it'd be great for metabolic diseases if we could do that. Now, the interesting thing too is if we go the other direction, go out of DRX to SRX, can you guys think of anything that would be good for?
Hibernation.
That's what they do. Yeah, there are the Ocala lab. They sneak up on hibernating bears and
no shit.
Yeah. They don't necessarily sneak up on them. I think they're
but they do like actually. Okay, because the whole time you're talking about this, like I was talking keto sleep, and that's what I thought it like. Is like.
And that's what the study came out within the last 6 months or so about from the Ocala lab doing a hibernation study with bears, and they've done. I think they did they did an omics study with it, which is super interesting because they've gone that direction to look at the more, it's strictly data science, bioinformatics stuff that they've looked at in these hibernating bears. But I was like, how cool. I talked earlier about, I don't think I could kill a rat, but I could muscle biopsy a bear.
I could totally see you like after, you take a postdoc there and Zach will be the one sneaking up on the bears. Got him!
So yeah, hibernation, the super relaxed state is very important for hibernation.
Okay.
Yeah. But just in humans, think about what kind of clinically even what's important to keep metabolic rate low.
Like at when is it important to keep metabolic
so like
rate
if
low?
you have a patient come in who has something going on with them, but we want to keep like I think about a burn victim, right? You want to keep their metabolic rate low,
sepsis,
right?
and somebody who has like
but
an
even,
extreme infection.
Even I'm not going to remember it now. Age-related muscle wasting, right? Like they're just muscle wasting in general. You want to keep that metabolic rate low because if it's increasing, they're going to that muscle wasting is going to snowball. So if you can shift the other direction and keep ATP turnover relatively low, you can help with some of that stuff too.
That made me think of something too. So Reed, I'm sorry, I keep jumping in really quickly. I know if you have questions.
No,
you just slapped me.
I'm just learning, just soaking it in right now.
So you talked about aging.
Yeah.
Now, and I know you're in an aging lab. Is one of the things that you guys are looking at, can keeping myosin out of this super relaxed state play a role in keeping muscle active to the point where you won't get atrophy at the same level? Like, are those experiments being done?
That's a good question. And it's hard. I always think of the difference between species, right? Like, this is something that's kind of difficult to translate to. The experiments are fine. You can do this in humans, but what I see in rodents is not necessarily what's going to happen in humans, right? Yeah. But in our lab, we might get into this in a little bit, but there have been 2 other studies out of our lab that have looked at super relaxed myosin in using 2 different models. We, 2015, a biophysicist in our lab did a overactimized model, but a few years ago, the person I took this research over for, she did an aging. I guess we've done 3 studies. We did an aging model where she took young and aged, adult and aged, doesn't matter, male and female, young and old mice, and went through the gamut, right? Like, that's a lot of data. That's a lot of fibers. The male animals, if you compared young and old mice, the parameters of relaxed myosin, no difference. But the females, so these aged females are ovarian senescent, so they are post-menopause, not producing estrogen anymore, estradiol, and they had significant differences between their SRX lifetimes. The aged females had shorter SRX lifetimes, meaning they're turning over ATP more rapidly in the SRX. And there was a trend that there would be more myosin in the DRX in those female mice, but nothing happened with the males. So to me, that says that there's something going on with the role of estrogen in regulating relaxed myosin. But it's not an aging thing. It's an estrogen thing, because if it was just general aging, you would see something happening in males, right? Yeah. And to further put a stamp on that, the overactimized study, the overactimized mice that they did was a few years prior to that. The mice that were overactimized had shorter SRX lifetimes than the sham operated or the control mice. But when they treated a subgroup of those overactimized mice with estrogen, those levels went back to sham levels. So that underscores the relationship that relaxed myosin has with estrogen. There's some kind of regulating role that estrogen has in there. Now, if you fast forward to now with my adiposity study, to this point, I don't think that there's been any male-female comparisons. Lean from my lab did the young and aged male and female, but she did not compare the male and the female. It was just within sex. So I took my study to the level of a 2x2 ANOVA, where I'm comparing diet and sex. And oddly enough, there's no the adiposity condition where these animals were fed a high-fat diet, there was no effective condition, no main effective condition on there. So the increased adiposity doesn't seem to have an effect on the SRX. However, sex did. And there were sex differences between these male and female mice, or male and female rats more so than the mice. But there are sex differences there, and that underscores, again, the relationship of estrogen with the myosin. However, you have an overoptimized model, you have an aging model, and in those animals, there was a destabilized SRX. But in my adiposity study, the low estrogen state is the males, right? Compared to, these are just young, healthy animals. The low estrogen state is the males, and they have a stabilized SRX, so their SRX lifetimes are longer.
So,
right? So it gets complex when you're looking at 4 different variables, and we just need to keep studying estrogen and what role it has in this, because now I'm thinking, what role does it have if in females this happens, in low estrogen state females this happens, but then I have low estrogen males and the opposite happens? What else is going on there? What else is involved? And I don't know.
Could be other hormones.
Oh, wow.
Other hormones, yeah. And I looked at the study I'm writing up now is on, I used a transgenic model of mice where we genetically ablated a receptor on muscle, an estrogen receptor in muscle, to see if these mice that can still produce estradiol but can't signal to the muscle, if there's a difference there. And I got p-values of like 1. So no.
For the listeners, a p-value of 1 means there's no significance,
Yeah.
and nothing
so-
happened.
Yeah. So the way I think about p-values, this might be not in the scope of the podcast, but a significant p-value is less than 0.05. And what that means is that if you do this experiment 100 times, if the thing that you are looking for happens less than 5 times, that is not by chance. There's a significant thing that you're looking at. If it only happens 4 times out of 100, there's a significant difference there.
Yeah.
That's a matter of, it's not a matter of chance.
Okay.
But
Stats.
Yeah. So if you have it, if you see something that happens 30 times, your p-value would be 0.3. That's up to variability. That can happen randomly. But if it's less than that, it's not random, or less than 0.05, it's not random. But anyways, so yeah, there's some regulation that's happening down the line here, and I think the people who are going to research this in the future really need to hammer in on what are the sex hormones, how are these sex hormones related to this, and how are they regulating the myosin states? And I don't really know. I don't know who else is studying that, but there's something happening there, and it's important.
But so we don't know though if you said it's not aging specific, and so. But do we know the answer to the question if the myosin state plays a role in like maintaining muscle mass, so like atrophy? Because I'm just wondering, like, if you were to have a more active myosin, you'd have a more active muscle in a sense,
Sure.
which should help you preserve muscle mass. I'm just thinking of it in terms of like a muscle mass question. You know what I mean?
So specifically, you're thinking myosin, like the state of myosin relative to muscle mass?
Having just a more active muscle. Like if that could play a role in preserving muscle mass with age, because people as they age lose so much muscle.
Sure.
And then is there a difference between type 2 and type 1 fiber, the rate of disordered relaxed state and the SRX life, and with age, we see a shift from type 2 to type 1. I'm just thinking of it in terms of like, you're talking about this metabolic aspect, which is huge, but could it be even bigger where we talk about old people lose all this freaking muscle? And then if there is a difference in type 1 and type 2, like, that should push people, or just in general, like that should just push people to build muscle.
Right. So,
which we're all about on this podcast. So that's why I ask.
Yeah, right. Gains. So, gains. As far as the aging question, I don't really know. The activity. So, I know that increasing muscle mass will increase muscle thermogenesis. Mm-hmm. It's just if you have more muscle mass, it means that you have a larger, more metabolically active tissue.
Yeah.
Right.
And you literally have more myosin. So
literally more myosin, but also more mitochondria. Right. So you have powerhouse.
So you just talked about that.
More mitochondria means more ATP production, and with increased ATP production, you get more ATP utilization. That is a thing that happens. Just because if you're producing ATP, it's got to get used, right? That's how this works. And so larger muscle, more mitochondria, more ATP production, more thermogenesis. And that's, to me, I got to take some notes because this might be something I can do. I'm thinking like it's a really good question, because if you look at, like we were talking about this, there's this complex relationship between these variables that I'm looking at, and I'm kind of wondering how much mitochondria matters to this now, because if, again, more mitochondria, more ATP production, does that relate to what I'm seeing in my data, where I have some questions about what's actually happening, and we can make some assumptions on heat production from mitochondria and ATP production. To answer your question, yeah, I think the size of the muscle is directly related to yeah.
Makes sense to me.
Yeah.
Now you're going to be dedicating a whole career.
Well, and that's so. I'm coming up to the end of my research time, right? I have essentially 3 studies that I've done: the mouse study, the rat study, and then I have this transgenic model that I've done. But I feel like I need more, because I have too many times where my answer is I don't know, and this might be. I have a shelf full of frozen muscle in my freezer, and this might be something that I can actually do, is measuring mitochondrial biogenesis in these muscles and comparing it to the known relaxed states that I already have from the opposite, the contralateral leg, right? So this is something that I probably can do in time for me to write about it in my dissertation.
Don't do that, Zach. You're going to stress yourself out way too much.
Hey man, I got 6 weeks when that baby comes.
Oh yeah,
lots of time. Yeah.
I'm not gonna. Emily will be home too. She can take care of the baby, right?
Yeah. Daddy's got an experiment to run.
I'm curious what you want to do with all this research you're doing. Like, what's your ideal career you'd like to get into? Is it going to be like sports, or what are you looking to do with all this?
That's another great question. I don't know.
Because you're almost ending your time with your research. So what's your next step, or what would be your ideal situation?
So
what do you want to be when you grow up?
Yeah, what do you want to do? Astronaut?
I've told Emily that I'm going to go to medical school.
So, hell yeah. follow my lead.
So, my goal initially with the PhD program was to stay in academia. I wanted to teach, and that's how it started. Is I was teaching labs at Mankato, and I enjoyed it, and I thought I was good at it. And if you can find something you like and you're good at it, like, go chase that.
Yeah.
Well, turns out that everybody wants to do that. They want to get a PhD and be a teacher and not have to do any research. Some people really like the research. I like what I do. I like my lab that I'm in, but I don't think of research as my ultimate goal. And I think part of that is research like this. Bench work, probably not my thing for forever. I just don't think I necessarily have the type of, I don't have the big ideas, right? Like to run a lab, you need to have big ideas,
and you got to just constantly beg for money. It's like a full-time, like, how much money can I get? Like,
right. And so I don't have the desire to constantly write grants. There is a job that there are jobs where I would do that because of the nature of the job, like VA researchers. They have to write grants to get funded to do the research. I'm all in on that. I will do that for sure if I could get hired to do that. But again, you need ideas. And in this world, I'm not necessarily the idea guy. I'm the doer. If you have an idea, I can answer a question. I can do the thing, but you need the question first before I can answer it.
Yeah.
Oh.
You get the creative mind in terms like how to come up with the answer.
Yeah.
There.
Right. So
there's a lot of pressure to come up with questions in academia that at a certain pace, like you have to publish a certain amount of studies. You got to get a certain amount of money at these high level research institutions like you're at, like your PI, if she wasn't getting these $1 million grants, she'd be out of a job.
Right.
And her job depends on the research.
She's on one of her grants. She's on her third renewal. I think she's had this grant for over 15 years. You have to renew it every 5 years, and she's on her third renewal of it. So she's pumping out research like it's her job, and she's doing it. She's a very prolific researcher, and she gets incredible people to work in the lab and pump out research studies that are important to the field of muscle physiology. And I would love to be able to contribute to that. Maybe I am with these studies that I'm doing. Part of that, it's so early on in SRX research, especially in skeletal muscle. It's so early on that some people might call what we're doing foundational. But I think a lot of what we're seeing now is, okay, Zack found these things. We're going to do a similar study and compare our work to Zack's. And what that's going to end up with is more questions than answers, because their data are going to be a little bit different than mine, and that's just going to raise more questions rather than like, yes, this is exactly what's happening in muscle, which is great. It's cool to be on the front end of this, but
that's wild. Like you're pioneering this like mechanistic area of study. Like that's so awesome.
Well, and so this is happening in parallel with cardiac muscle. So there's 3 types of muscle in the body: smooth muscle, cardiac, and skeletal muscle. And smooth muscle is kind of off on doing its own thing because it's involuntary. You don't have any control over that. But there is super relaxed myosin in smooth muscle. They've measured it. But in cardiac muscle and skeletal muscle, they're both striated, meaning that they are these super organized muscle, these super organized tissues that are like a sarcomere is the basic contractile unit of a muscle, where that's where those molecular proteins live, and sarcomeres are stacked one after another, and you have thousands of them in one fiber. And in striated muscle, cardiac and skeletal muscle, that's why it's striated. It's these sarcomeres getting stacked on top of each other. Well, skeletal muscle SRX research is being done in parallel to cardiac muscle SRX research. However, the cardiac stuff is moving way more quickly than skeletal muscle. So cardiac muscle, they have taken the SRX research and actually have a drug on the market to treat dilated hypertrophic cardiomyopathy because of the research that's already been done in the cardiac sphere. And it just moved so quickly, so efficiently, that they've already been through, in 15 years, they've already been through the foundational studies, the clinical trials, wow. And there's a drug out there. It's called mavacamten, and it stabilizes super relaxed myosin in cardiac muscle. And if you think about what happens in stabilized SRX myosin, they are remaining in the SRX longer, or there's more myosin shifting out of DRX to SRX and staying there longer. And in hypertrophic cardiomyopathy, you want that to happen because you do not want myosin in the DRX pumping out, hydrolyzing ATP more quickly, or you're just going to get more hypertrophy.
Interesting.
Right? So that drug is on the market and it works.
So this drug specifically induces that stable SRX state.
Right.
Wow.
So Reed asked me what I want to do with my life.
We are so similar, Zach.
It is. It's not that. It's not that.
Everything you just talked about, not that.
We're just gonna go through the line.
So what I have found over the course of my time in the PhD is I still enjoy teaching and I'm still good at it, but I don't need to be an instructor, a formal instructor at a university to be a teacher. And having worked in medical devices and now getting the PhD, I have found careers that I can do where I'm still going to feel like I am teaching someone something. And if that's my tool bag is an educator, I can find jobs like that. And what I'm after is some companies have product managers. I think that's probably tier one of what I want to do is product management, because I can get my hands on devices and I can learn how the thing works, take it apart, put it back together. If I can do that, great. And then I can teach other people how to use it. And then I can work with engineers on how to make it better and just work through the whole life cycle of a product. And for me, that's very intriguing to me. The hard part is I'm not a marketer, right? Like that's a marketing position at most of these companies. But if I can get in the right one, specifically medical devices, I can leverage my experience to use what I've learned. You know, the PhD is not about, sure, you pump out some research, you get published. That's great. But a PhD is more about learning how to think. Mm-hmm. And if you can think critically about a problem, ask some questions, think about how to solve the problem, answer your questions, and then actually do the thing, and you're going to hit some roadblocks along the way. You got to troubleshoot. That's what the PhD is really for. And that's what I'm learning and have learned how to do. And that's the type of career that I can find myself in, where we have this idea, let's execute, let's run into problems, let's fix the problems, and then move on from there. So product management is very much like that. But I'm also interested in some clinical roles. Clinical research at Medtronic is another spot that I'm interested in. And then there's a friend of mine works for a company it's a global company, and they have these what are they called? A field clinical specialist, where you are in the field, and this particular one that I have considered applying for is it's a cardiac device that gets implanted in an OR, and as the field clinical specialist for that device, every time that device is implanted, you have to be in the OR present to monitor.
In case something goes wrong.
In case something goes wrong, in case there's questions, and you just have to be there as the professional on the device. And that sounds super cool to me.
Yeah.
And that whenever you hear field in a job title, it's a little freaky because you got to travel a lot. And for me, that would be great. I would love to travel all over the country, all over the world doing a job. But I have a baby on the way.
Yeah, it's tough.
I can't do that. So this one in particular is a Minneapolis-based role. So I would just stay in the Twin Cities, driving around from clinic, hospital to hospital, whatever. And so somewhere along those lines of working with a specific product or group of products, learning how they work, teaching other people how they work, and making them better is really, if I can find a job where I can do that, great.
Awesome. Yeah.
That's interesting to hear you explain what you learned in a PhD and apply that to jobs. I've never thought of it that way.
I was hoping you'd say work with the Vikings, get us to a Super Bowl, win the Super Bowl, not just get us. But I mean, those ones are cool.
Yeah.
Those are cool too.
I applied for a job with the Twins as a baseball guy, as a data guy. I applied for a sports scientist position with the Twins, and I was super excited because I sent in my resume, and a week later they got back to me and they're like, yeah, let's move you on to the next round. And the next round was a project that you had to do. It was like a 6-question assignment. 3 of those questions were biomechanics questions.
Oh, hell yeah.
Yeah, I was like, hell yeah, I can do that. And the other 3 were coding questions in R. Well, you build up some tools in your PhD program to solve some problems that you got, and I've really learned R to have in my tool bag for the next step in case I find I'd take a statistician job. I'd be a biostatistician at a hospital or something. I can do
R
that.
is stats software, by the way.
Yeah.
So would you have used SPSS at Mankato? Does that ring a bell to you at all?
No. That was many
I feel
years
like
ago.
stats is just grad school stuff. I didn't do any stats undergrad.
I don't recall that.
You did?
Anyways, yeah. So it's just coding, right? It's coding any other language, Python, whatever, but it's specifically meant for statistics. And I've gotten good at it, and it's just a nice tool to have.
That's huge because stats is the worst part of doing research.
And the thing about stuff like that too is it's a puzzle, right? To me, it's a puzzle. And if you can, I like puzzles, so I can go through my coding and I can figure out the pattern of how this code is supposed to work and then execute. And so that kind of coding language is right up my alley. So anyways, I did this assignment, biomechanics and statistics, sent it to him. Well, it turns out there's a staff scientist in my lab who got his PhD at East Carolina. I was randomly looking on LinkedIn, and I saw a guy who works for the Twins in sports science, who also got his PhD at ECU. And I turned my chair around and I go, "Hey, Luke, do you know this guy?" Sure enough, they knew each other, got his phone number, and Lab Luke, not that Luke.
Oh, I was like, nice.
No, I don't know
a lot of people.
When we talk about Luke in my lab, we
Lab Luke.
Yeah.
Lab Luke.
So he got me in touch with this guy who works for the Twins, and we talked about the job, and it's okay that it didn't work out, but it would have been super cool that it would have been a sweet job. Byron Buxton would have known me by name.
Oh,
right?
Keep us
so cool.
Me and Carlos Correa could have been friends.
keep us in mind when you eventually work for a sports team.
Yeah.
Hopefully Vikings.
Hopefully Vikings. Yeah. So timeout.
Are you once you're done answering the question?
There was something else I was going to answer that doesn't need to be in the podcast, but you had asked me sports team stuff.
Was it? I was just hoping you were going to work for.
I
thought you were going to work for a sports team, just because I know how passionate you are in sports. So that's definitely didn't think you were going to go into medical sales.
Sure.
Like a project manager.
What was I going to tell you? It doesn't matter. But I applied for a job with the Twins. That's kind of cool.
That's awesome.
I remember what I was going to tell you too.
Oh,
so I drive to you every day. I work in my lab every day, and last week I was walking from my parking spot to the lab, and I just bumped into some guy named Co Kieft. Do you know that name?
Co Kieft.
Do you know Co Kieft?
No.
So Co played at the U. He's a tight end at the U, and now he's a tight end for the Buccaneers.
Oh,
so I'm walking into campus. He's very like
tall and handsome.
Well, he stands out in a crowd. He's got a distinct look. He's got red hair, and he's got long hair and a beard, and it's red. And like you can pick him out in a crowd. And so I recognized him right away on whatever day it was, Wednesday. I saw him on Wednesday. We were walking opposite directions. The next day I saw him again. And this time we were walking in the same direction, but he was with somebody else, and they were walking in front of me. And I hollered up to him. I go, "I saw you guys yesterday. You were moving better yesterday. Are you sore or just not excited about where you're going?" And they both look at me and they go, "Both." So I talked to them for a few minutes, which is pretty cool. I mean, he played tight end for Tom Brady.
Did you say you knew who he was?
No.
Or you didn't?
Is he still on the roster?
Uh-huh.
Oh,
yeah.
Nice.
Yeah. Kieft. Co Kieft.
Co Kieft. That's awesome.
Yeah, it's pretty. That's what I was.
That's
When you said Vikings, that's what made me think of it. I'm like,
Co Kieft.
Yeah, Co was a gopher.
All right, let's get into Reed's segment. My segment. Reed's Reddit finds. I looked up metabolism questions, and I just thought since we got the master here and we got Dr. Luke, let's just get both your brains and just see how we can help our listeners here. All right, so for the first Reed's Reddit finds. God, it's hard to say. You gotta say it a couple times fast.
Reed's Reddit.
Is it worth getting a metabolic test? Like, this is what one of the people on Reddit said. Like, is it worth it
to know your kind of resting metabolism?
Yeah, like is that?
I guess you don't know what they're actually asking. Yes, with some caveats. There are a ton of companies out there that are out there just to make money, and they will push bunk science. And in this day and age of internet, social media, it's hard to parse through those kind of things, right? So if we're talking strictly metabolic tests, it's worth it. However, the devices out there to measure your resting metabolic rate if you go to the doctor, you go to your doctor and get one done, you're probably going to be using a good device. There are devices out there that aren't as good, and if you want to know, you have to be hooked up to a mask, and it has to be a breath by breath measurement. Otherwise, you're not going to get any kind of accurate data. If you don't care about that, and you just want this super ballpark estimate, we got equations that we can give you. And that's, you plug in your height, your weight, your whatever physical activity level, and we can get you an estimated metabolic rate.
But it's not going to be very accurate. It's just kind of a guess. Yeah, because everybody's so different. Right. I used to do testing as well, and everybody was so different. So those generic ones just get you somewhere, but it's not going to give you your accurate reads.
So you were a metabolic specialist at Lifetime, right?
Yep.
So I did that for a spell,
right.
Yeah, just for a few months in between after I graduated from the master's and went on to MGC. So that device, the VO2000, or I don't remember what Lifetime called it.
Oh gosh,
doesn't matter.
What it's called, yeah.
So those devices that company that built those devices was owned by MGC, the company that I used to work for.
Okay.
And so that's the first thing that comes to my mind when I'm talking about, not necessarily like bunk science. That's not what that is. But that is not the kind of device that you want to use to get a super accurate metabolic assessment. It's fine for the gym, but if you want something better, I think it's better to go into a clinical setting, or there's companies out there like DexaFit. You guys ever heard of DexaFit? Mm-mm. So it is a performance gym, but they also do some rehabilitation and stuff like that. And shameless plug, MGC, they're a client of MGC, which is why I know about them. So they have fully functional breath by breath analysis where you can literally measure breath by breath. So like our cart at Mankato was a Parvo cart. That has breath by breath analysis, but everything that we did was 15-second averaging. Again, if there's a Parvo cart near you, you can find one. Universities often have them. If there's an exercise science program, that's great. That's exactly what you need. But if you want to the breath accuracy, same thing. You'll find them at gyms, or not gyms, sorry. You'll find them at universities sometimes, but those are really meant for clinical settings. So if you got a doctor that will do that test for you, that's kind of what.
Okay, awesome. So like if you're really interested in it, the ones at the gyms will just give you some generic stuff, but go to more of a clinic setting if you want your exact. Mm-hmm. Okay.
Yeah, and if you're in good shape and you're a healthy person, I think something like what was at Lifetime, I have kind of an issue with that because of the way that the breath is measured more than anything else. Some people might be better than others, but I think as you get to more of a scientifically involved facility, you're going to just get better devices.
Yeah. Okay, that makes sense. And then question number 2 on Reed's Reddit Finds is, how do I increase my metabolism? That's a very basic question, but I think that's one of the most important ones.
Run.
Just run.
So I have a list of some things here that are, moving is really the biggest thing, right? Like exercise, and both resistance training and endurance training will increase that. Sometimes it's genetic though, right? Like sometimes you can only get so far. It's a genetics thing. Like some people can, some people can't. But a healthy diet and regular exercise are going to be the best way for you to increase your metabolism.
So one of the things that I try to push people towards in terms of like increasing metabolic rate is people get so hung up on cardio. Like, I'm just going to go do cardio and it's going to burn the most fat. But in reality, and like what you're talking about, I'm really glad we had this conversation because now I can throw another piece of information at people of like, if you build muscle, like you need to resistance train. If you build muscle, you've got a larger capacity to burn energy and metabolic rate's going to go up because, you mentioned how much 40% of your energy expenditure is used by the muscle. Well, if you have more muscle, that muscle is going to require more energy and more metabolic rate. So like just a plug for resistance training,
Right.
because I think people do it so little, especially as people age.
Right.
You know?
And, you know, I'm sure, especially women, perimenopausal, postmenopausal women, resistance training could not be like, resistance training is probably just as, if not more important than endurance training for middle-aged women who are experiencing those changes in estrogen levels, right? As soon as they're postmenopausal, the prevalence of osteoporosis skyrockets. And one of the best ways to combat that is resistance training. You're a doctor. Am I making that up?
Yeah, no, absolutely. So you do see that. And then when you look back at the Women's Health Initiative study that was done in the 90s, and like they were essentially studying, should we be replacing hormones for individuals in menopause? Because what happens in menopause is like, so you have this period of time from when you start menstruation until, careful, Zach, don't spill that coffee. Okay, you can spill it all over. So you have this period of time from when you start menstruation to when you go through menopause. And every month during that period when you have your period, you've got this like oscillation of hormones. And then when you go through menopause, that oscillation doesn't occur anymore, and you have this decline in estrogen at baseline, or estradiol. And estrogen plays a role in stimulating or increasing bone mineral density. And so all these people in menopause have osteoporosis, like you're saying. And where was I going? Oh, Women's Health Initiative. Yeah, see, this is what happens, Zach. So Women's Health Initiative study is like, oh, you know, not only are people getting osteoporosis, but we also are seeing like these issues with menopause, like hot flashes and all that are hormone related. Should we be replacing that estrogen in these individuals? And so they did hormone replacement therapy, but the study was stopped because they're like, oh no, this is causing breast cancer. And it's a classic case of just horrible science. When you look back at the data, it's just as bad as like the science we were talking about in one of the other podcasts is about meat. Like, oh, meat causes this. And they do just like the cardinal sin in science of they report relative risk without reporting absolute risk.
Yep.
And so what they did is they found that for every 1,000 patients that were taking this hormone replacement, it was 5 of them, actually 5 out of those 1,000 developed breast cancer, versus 4 out of 1,000 developed breast cancer that were in the placebo group. And so absolute is comparing those two values. That's a 0.01% difference. That's nothing.
Right.
But relative, you know, that's a 25% increase. So then headlines are like, oh, hormone replacement therapy causes a 25% increase in breast cancer. So then like all these hormone replacement drugs and like the idea of doing that was pulled from the shelves. It's not done clinically because there's all this concern, which I think is such a problem. Decades later, you know, we're talking about the benefits that estrogen provides, not only to muscle, but I mean to bone, but potentially with muscle as well.
Yeah, it's the importance of estrogen. We specifically study estradiol in my lab. It can't be overstated. The importance of estradiol to skeletal muscle. It is protective. There's this regulation. It is so important to have regulating or regular production and binding of estradiol to skeletal muscle. And if it's lost, we see this whole cascade, this whole host of issues that happen in estrogen deficiency. And I mean, by adding that back in, I talked about that study earlier. Just this one little tiny component of skeletal muscle physiology, by adding estrogen back in after a bilateral ovarectomy, returned the SRX back to normal levels.
So in other words, like that's me asking you to like summarize like what that means again.
Yeah. So the overectomized mice that do not have circulating estradiol, they had a shorter SRX lifetime, meaning they're hydrolyzing ATP much quicker. This is where things get tricky because I said that that's good. Increasing ATP hydrolysis means higher muscle thermogenesis. Now, that in itself, in that scenario, an overectomized mouse having a higher rate of ATP hydrolysis is negative because that mouse is overectomized, right? So by adding estrogen back to that mouse and that those values are returning back to the sham level, means that just simply by treating the animal with estrogen and having it back to normal levels, that really focuses in on the importance of estradiol, whether it's regulating or through treatment, right?
To just get
back to normal.
Yeah.
Back
muscle function.
Yeah.
The reason I asked you to clarify is because I think that's going to be an awesome little clip. I think that was a really interesting point you made, but I didn't want it to end with SRX, DRX, because then people wouldn't know what you're talking about.
Sure.
So yeah, that's why I had you do it.
Got it.
All right, question number 3. We basically just talked about it right here. How do I speed up my metabolism after 40?
Oh, with aging.
With aging. We kind of touched upon it a little bit.
I mean, the process of aging at 40, you're not losing a whole bunch of muscle. So really, it shouldn't be any different, right?
Right,
I mean,
right. Especially in men, right? It's at 40, it's still relatively, if you're recreationally active at 40 as a man, it's fine. You're starting to approach perimenopause as a 40-year-old woman. 46, 47, 48, somewhere in there is when those perimenopausal signs start happening. It's really a time to start thinking about taking into account your health, your diet, your physical activity levels. And again, we go back to the previous, it's the same thing. Keep resistance training, endurance training, clean, healthy diet. I'm not talking about dieting. I'm talking about just eating and moving. And I don't know how you guys talked about meat in a podcast. I'm not a huge proponent for dieting. I think it's kind of a sham, because you can really fuck up your metabolism with all of the yo-yoing. And I've never met anybody who's on a diet and doesn't yo-yo. Exactly. It can be so hard on your body, and it can increase inflammation, and that is worse than how you were before you started your diet. I would rather not diet and eat normal and not have any inflammation, right?
Yeah. That's 100%. We started this podcast just talking about how nutrition research is just flawed, and don't diet. You know what works best for you. Just don't overeat.
Yeah. So it's about portion control. And I mean, I like to tell people added sugar is really is kind of what's not really good for you. However, if you like sweet stuff, you know what counteracts added sugar? Exercise, right? If you can combat some of your caloric intake with a relative amount, a relative amount of physical activity,
got a little more room for sugar.
Right. And that's your body's going to process table sugar the same way it's going to process the sugar in a banana. Your body doesn't know the difference. So if that's something that you do, you just have to normal exercise, normal diet. And if you want to try the Mediterranean diet, by all means, go ahead. But you got to be careful if you're going to switch and just completely revamp it. Mediterranean diet probably is not a great example, because that's probably the best one of the bunch.
In medical school, that's
number one.
Only thing we talk about in nutrition or about nutrition in medical school is just recommend the Mediterranean diet, not why we recommend it or any of the research. It's just that's always what's
interesting to tangent here. Sorry, Reed. Sorry, Reddit.
He's used to it.
The thing about me personally and the Mediterranean diet is that it's lots of fish, right? Lots of grains, quinoa and rice, and just grains like that. But it's lots of fish, and fish are fatty. And I'm a proponent of make sure you're getting your dietary fat. Mm-hmm. It's important.
Yeah.
And if you are ingesting the right amount of dietary fat, your body is still going to break that down into glucose and use it as energy. And gluconeogenesis, right? That's where that comes from. It's taking fat and breaking it down so that you have the energy that you need, whether that's coming from carbohydrates or from fat. Your body can break that down into what it's going to need. So that's the nice thing about the Mediterranean diet is it's somewhat higher in fat, depending on the fish that you eat.
I mean, traditionally it is a healthier diet,
diets are a sham.
We take your stance on that 100%. before you go into the next question, Reed, so exercise, and you talked about exercise specifically having an influence on the state of myosin in a beneficial way. Now, I know that there's some research when I was poking around a little bit into EPOC, excess
post-exercise oxygen consumption.
Excess post-exercise oxygen consumption, which is essentially like after you're done exercising, you still have this elevated metabolic rate to restore all the energy that you just utilized. And I saw that there was some research into the state of myosin on EPOC.
Yeah. Did you read that Cook paper that was in Metabolism about the BMI study and the piperine?
That's the one I'm talking. Yeah. So that was going to go to my next question. Yeah. And so what they found is that if you did, it was like 30 minutes of walking had a certain level of EPOC, which again is just like raised metabolic rate after walking. Well, there was a higher amount overall of energy utilization if you broke it into little segments.
Right.
Maybe due to this myosin state of like, oh, we shouldn't be sitting for a prolonged period of time. And like, yeah, so I just wanted you to speak on that a little bit.
Yeah. So they also talk about how fidgeting is can be beneficial to that. So we were talking about NEAT earlier, the non-exercise activity thermogenesis, and they're like fidgeting. Like you get fidget spinner is not really the thing, but you see these kids at schools that have the little fidget desk where they have there's a swing that they put their feet on and they can fidget their leg. Like that's based in science. That's not just people like get this kid to burn some energy. That is based in science, that having this kind of these tools at your disposal to be able to maintain some level of activity with intermittent spurts throughout the day can keep that EPOC high longer, and it keeps your thermogenesis higher longer, not necessarily to the extent of what exercise will do, but just maintaining some level of elevated activity that's more than sitting will keep your thermogenesis up, keep your metabolism up, but it doesn't feel like you're working out, which is, I'm sure, a great thing for a lot of people out there who, like me, I'm like, I don't want to work out, but there are movements that you can do that are still contributing to these changes in thermogenesis that are positive for your overall metabolic health, and it doesn't take a lot. It's just move.
Exactly. It'd be interesting to see if like people that fidget a ton, like if we looked at Reed's DRX, where she's like Allie, she's super still. Like if fidgeting and that increased metabolic rate, if like the stuff you're studying it plays a role.
So shameless plug for my lab too. The Lean, who did this study, this work before me in my lab, she was the first person to study to measure SRX in human skeletal muscle. It had never been done before, and in 2020. The study got published. So like 2019, she had muscle biopsies from a collaborator at UMass, and they were the first people to measure the SRX and DRX in human skeletal muscle fibers, which is pretty cool. So
and that was so recent.
Yeah. Because again, they see it started it's really started in parallel with the cardiac and the skeletal muscle, but the cardiac took off so quickly that people were like, we'll put the skeletal on the back burner for now. And we work in a skeletal muscle lab, so we did not do that.
Yeah, you're not putting it on the back burner.
But that's the biggest thing is just move, man. Like I can't say it enough. I can't just move. And you don't need to go out and do high intensity interval training. If you like to, great. It's awesome. High intensity interval training is awesome. It takes 20 minutes. You can be done with your workout in 20 minutes.
Yeah.
But if you don't want to, walk your dog. That's going to elevate your muscle thermogenesis. Just walk your dog. If you don't got a dog, walk your cat. I don't care. Just move. Just move. There's people out there walking lizards these days. Like they're just trying
to ramp up that thermogenesis.
Right.
Is it better, in your opinion, to space those out, like what that lab was potentially suggesting? And like, how does that impact today's society where we're just sitting all day long? Like that just adds some more science to like get up and move for a little bit. and the influence that that's having on muscle thermogenesis. This mechanism, I'm just really happy that you brought this up because it relates to all these like
You brought it up.
Well, no, I'm bringing it up and relating it to like the mechanism that you're teaching us.
So like so I think you're right. I think by spreading out those short bursts of non-exercise activities is better than doing it all at once, because what are you going to do after an hour workout? You're going to shower and then go sit on your ass. Like that's what I do. I'm just going to go sit
every
right?
time.
But if you can take a 10-minute walk at work and then go stand at your standing desk and then a couple hours later go do it again, you're going to see repeated bouts of excess EPOC. You know, the easiest way to think of EPOC is like you hear people talk about it, the afterburn, right? That's why you're still like, that's why you do a workout, you go home and you shower, and when you get out of the shower, you're still sweating. Yeah. That's EPOC. That's what's happening. You're still consuming oxygen at the muscle level, elevated relative to your just your basal state.
That always happens to me. I always thought I was just a sweaty person.
No.
Look at me.
Yeah, you're still just like ramped up.
So that is the afterburn, the EPOC that you are experiencing, and that if you can do that for 10, 20 minutes at a time, 3 or 4 times a day, it doesn't take a lot, but you have that EPOC elevated multiple times, so you're just going to have this longer overall afterburn than you would after a long workout. It's just because if that EPOC lasts 30 minutes per bout, but you only do one bout, compared to 3, it's just, that's simple math.
That's
people.
science. Yeah. I love that.
All right, question number 4. We kind of been like, these all kind of segue to each other, so it's kind of great. It flows really nice. But can you really mess up your metabolism from eating too little? Or is that a myth?
No, I think, I don't have any science to back that up, but I mean, it depends on how extreme you go, right? Because if you are not nourishing your body, your muscle is going to atrophy. You're going to end up wasting, right? You're just going to waste away. And obviously there's a fine line there where I don't like this, but if you want to lose weight, caloric restriction is necessary, right? Doesn't need to be a crazy amount of caloric restriction again if you're moving, but if you take that too far, your muscle is going to atrophy, and it's just you're better off not doing anything at all and just staying where you are. Yeah. You know what I mean?
I just had another thing pop in my head. So I'm just obsessed with this SRX. So you asked about energy restriction and like hibernation as it relates to SRX. Like, do we know a potential like signaling mechanism? Well, of course we don't know that because this is just in its infancy, but like, I wonder what the mechanism would be to trigger your body into like starvation mode. Like when you go on these prolonged fasts and stuff, like, are you inducing in our SRX state decreasing metabolic rate? And then that would, answer the question in a different way of like, you're lowering your metabolic rate because your body thinks it's starving.
Yeah.
Oh, wow. And it all comes down to the muscle. Like that's just fascinating.
Right. Again, and I brought it up a couple of times. It just comes down to volume of the tissue in your body, right? Like that the muscle is so important to your overall metabolism because of that. And I think I would imagine that there's changes to the kinetic states of myosin strictly based off of your diet. Now, my study says the opposite, but these are rodents, right? These are relatively simple models.
But your study's more of like an overfeeding study.
Correct. Right. So the opposite of that.
Right. Which wouldn't, well, I guess you could say you're looking at the opposite.
So the reason I wanted to do that study in particular is because the original paper, the seminal paper that named the SRX, theorized that by an acute bout of overfeeding, an excess caloric intake will shift myosin out of the SRX to the DRX, right? And that bout is just acute, but I wanted to make it chronic and extend that and see what happens with a long period of time. But like I said, postprandially, you have an increase in thermogenesis after just the meal, but that thermogenesis is a little bit elevated. If you overeat, right? There's an excess caloric intake. And that change in thermogenesis is roughly what you would see if you shifted, like I said, 20% of myosin out of the SRX to the DRX. That's the change in thermogenesis that you see there that's roughly equivalent of, if that's actually the mechanism that's happening, why thermogenesis increases is because myosin is unfolding. That's what that's equivalent to. So I would think because the amount of myosin in each respective state, it's a proportion. It's out of one. DRX is 75%, SRX is 25% in my studies. That's how it worked out. But if myosin comes out of the SRX, it goes to the DRX. And I think, and because it's out of one, that means myosin and DRX has to go to SRX, right? The proportion has to equal one. So I would imagine if the opposite is true, where excess caloric intake makes the shift happen SRX to DRX, that the opposite would occur because of the nature of the variables.
You just dove down to like the depths of the ocean, came back up, right back up to answer that question. That was wonderful. Beautifully done.
Did I answer the question?
You did. Absolutely you did.
And then my last question we got is supplements or methods to boost your metabolism. Because like you'll see some commercials out there on TV that are like boost your metabolism, like metabolism boosters. And then there's also that study you're doing on increasing the DRX. Like, what are your thoughts on those?
On supplements?
Yeah,
supplements. I think supplements are a crock of shit. The nutritional supplement industry is an unregulated industry.
Yep.
They can tell you whatever they want to tell you, and it is not examined by any governing body, specifically the FDA.
Yeah.
They can say whatever the hell they want, and they cannot be punished for it.
Yeah.
They veil that in a way that is so clever, marketing-wise, that people believe it. People believe whatever these, like, sure, there's some testosterone boosters that are going to increase your thermogenesis, but you know what else increases testosterone?
Exercise.
Fucking squatting, right? Like,
I didn't even tell him to do this.
so
It aligns perfectly.
This is what I do to them all the time. I'm like, you know what else does it? And they just exercise. Unless you say collagen.
I've been a strength and conditioning coach I coach strength and conditioning at Mankato. I coached at the high school level. I've done it for years. I've always been the legs feed the wolf, because you have big muscles in your legs and in your butt, and if you can
booty cheeks,
those glutes, man. If you can grow those muscles, guess what? You're going to produce more testosterone.
You don't need no pill. Dude, the stuff with that supplement industry is wild, though. I did a lecture a few weeks ago on just like the supplement industry and what you're saying. And I was looking into some of the research on, there's a couple labs that have basically pulled a bunch of supplements from the shelves into different, in different industries, if that makes sense. Like testosterone, for example, they pulled a bunch of testosterone supplements and then looked at like, okay, what do they say is in the supplement versus what is actually in the supplement? And it is wild. Like most of the time it's, like
fillers and
well, it's not actually the amount that they're saying.
Put sawdust in there for
Well, there's a certain regulation that comes from the FDA where if you make a certain statement claiming that it's going to have an impact on your health, that is regulated by the FDA, but you can word it in a way where it's not a claim so that it isn't regulated. But if it is, then what they're required to do is list all ingredients, but they can list it at the bottom. If you ever look at the supplements at the very bottom where it doesn't tell you how much of each component, it's just a list of them. They do that because they're not required to list the quantity. The purpose of that was so that you couldn't plagiarize and take someone's recipe and put it elsewhere. But they use that to say, oh yeah, we have this component in ours, but it's probably just like a fraction, save money, and
just
it's
trace
a load
amounts,
of shit.
Yeah.
What did you call it at the beginning? A crock of shit.
Probably a crock of shit.
Yeah, it's a crock of shit,
Mike.
I will say that if you're looking for a supplement to acutely increase thermogenesis, caffeine. You can take a caffeine pill. That'll do it.
Oh yeah.
You might as well drink coffee though.
Yeah.
Right. Like
I was just gonna say that. I'll just crush some coffee.
Yeah. Yeah. And that's all of this to say, we haven't even talked about pre-workouts, man. Like,
Oh, I used to take those so much.
that,
Jack 3D.
yeah, man. Like, if you want those effects, drink a cup of coffee. You'll be perfectly fine. Like that.
That's what I started doing when I worked at Lifetime.
See,
It's like I
coffee's-
used to take pre-workout, and then all of a sudden I met this trainer that was Jack. He's like, I just take coffee and creatine. That's all I do.
I haven't taken pre-workout in years. And coffee, like the actual bean itself, has some really impressive.
Yeah, man.
You know,
I mean, it's a
benefits
fruit
to your body.
It's
Yeah.
technically a fruit,
Is it
right?
really?
Yeah, it's a berry, right? If you look at a coffee bean before it is a bean, it looks like a tiny little
and then you
apple.
just dry it out.
Mm-hmm. That's all. Chocolate's a fruit too, by the way.
He's a really good trivia guy.
Yeah, I mean, cocoa pods are like this big, and they have to like cut them apart, peel them apart, and then there's these beans in there that there's this fruity, fleshy stuff on the beans, and then those get dried and fermented, and then those become cacao nibs, and then those get mixed in with the milk fat to make sugar, or they get kind of melted down with sugar to make chocolate or whatever. So
Whoa.
it's a fruit. Chocolate's a fruit.
I thought we were
man.
learning about metabolism.
Zach's
Now I
gonna
know.
open up like a
Well,
shop.
dark chocolate has
Yes.
there's a ton of health benefits to dark chocolate, obviously in moderation. If you're eating chocolate and it's not dark, you're doing it wrong, but you're doing that. Yeah. It's caffeine too. I think there's trace amounts of caffeine in dark chocolate. So have a coffee and a little chunk of dark chocolate, go work out. You'll be great.
Boom, metabolic rate skyrocket.
Yeah. Yeah.
So exercise and caffeine.
Yeah. Don't take those supplements. They're not regulated.
If you
That's
want
crazy that supplements claim to boost you. Well, I suppose
Oh, yeah.
it's probably like one of the
Well,
hottest
watch TV
industries.
late at night.
Yeah,
You'll
yeah.
hear it all the time.
There was one at GNC that I took back in the day. It came in a vitamin pack with multivitamin and like a water X pill, and there was a thermo booster. I don't remember what they called it, but it made you feel like dog shit because they claim that it's increasing your heat production, and I mean, I don't know if it actually did that, but you felt terrible taking it. The multivitamin's great, but the rest of it was just god awful. Taking a water X pill that is meant to rid you of your water weight, that sounds like a great way to die.
Yeah. Let's just throw that in there.
What the heck, dude?
Yeah,
But
yeah.
I mean,
Wow.
like, it's such a hot industry that people make whatever hell claims they want.
Well, right, and you get somebody who's really gung ho into like, all right, I want to lose 25 pounds. I want to get my beach bod going, right? So they go to GNC, they get these packs, these vitamin packs, but on top of that, they get back into the gym and are doing regular exercise in whatever way. I quit going to the gym after I quit playing college football. I was sick of it. I didn't like it anymore. I was just burnt out, and I quit going. And then one day I was like, well, I might as well start doing this again. And just by training on an elliptical, I lost 15 pounds, right? Like that, if that's what people are after, you don't need all this extra stuff to do that. Regular
Just put in the
exercise,
work.
healthy food, and a little bit of motivation, right? Like it's difficult. I get it.
Discipline.
Especially it takes a lot of discipline, but I'm going to be 34 here. I don't have time to go out and do 2-hour workouts like I did back in the day. And
miss
I
those
get
days.
it. It's hard.
Yeah.
It's hard, but it's not hard enough to not do it.
Right.
And now I'm convincing myself maybe I need to exercise more.
That's what we're here for.
Yeah.
That was it for Reed's Reddit finds. Yeah, I appreciate that.
All right.
I got a Luke's Reddit find.
Yeah.
Okay. So we did an episode on this, and actually you reached out to me afterwards because it relates to your research. So the cold water immersion, everyone's doing it.
Yep.
It's like the thing. If you're not doing it, then you're a loser on social
Yeah.
media. And like one of the biggest claims is this is like the best method to burn body fat. Of course,
Gary
it's the
Brecka.
best method. Yeah, Gary Brecka literally called it the best method for losing weight, raising metabolic rate. Now, the claim is made because of how it activates brown adipose tissue, which you touched on, mitochondria and mitochondrial uncoupling and how that raises metabolic rate. But when you look into the studies that were done in Denmark, like they weren't actually asking that question when they did the research. And it doesn't seem to be that this cold water immersion really has much of an influence on brown adipose tissue, especially overall metabolic rate. So I want you to talk a little bit about that, because you did reach, you didn't specify, but you were just like, basically like, hey, I'm over here.
I really did. I think that was on Instagram. I was like, yeah, so
this happens to be an area of expertise of mine.
Well, I wouldn't go that far. So, Luke mentioned brown adipose tissue, which is so you have, I guess, kind of 3 different kinds of adipose tissue. There's brown and white and beige, right? In animals, there's really white and brown adipose tissue, especially in rodents that I study. There's a lot of brown adipose tissue. In humans, that brown adipose tissue is more metabolically active, but there's so little brown adipose tissue in humans that's probably why the people in Denmark who did that study didn't see the changes that either they were expecting or just the internet is expecting. It's because there's so little brown adipose tissue, except for when we're infants. And it's, as infants, it's so difficult to regulate body temperature that it's necessary to have the brown adipose tissue. So what happens in brown adipose tissue as far as thermogenesis goes is there's something in that tissue called UCP1. It's uncoupling protein 1. And what that does is it uncouples this process that increases ATP utilization, so it increases heat at kind of a disproportional rate. Right? Is that?
Yeah. So the way, and you explain it much quicker than when I did in that podcast. Yeah.
So with brown adipose, it would be great if we could harness that in human metabolism. It would be great, but there's just so little of it. And what happens more so in humans is the beiging of white adipose tissue that becomes kind of brown, and it just becomes more thermogenic. So I think what cold exposure. Fine. If you want to do that, what did Bob used to say to us back at Mankato? Bob would say, if you want to lose weight, go stand outside naked. And because shivering thermogenesis is a real thing, because what happens when you're shivering due to the cold, there's involuntary muscle contractions happening. That's smooth muscle that is contracting and utilizing ATP. So your thermogenesis will increase as a result of shivering. And related to my studies, the work that I do, that same paper, the seminal paper from back in the day that made all these claims sounds derogatory, but in addition to this acute postprandial period of increase in thermogenesis, cold exposure theoretically does the same thing. That would be a little bit easier to study than the diet stuff that we dug into. But the same thing has been said. The diet stuff is just much more of a hot button issue than the cold exposure stuff was at the time. But yeah, you take some animals that are exposed to cold, their thermogenesis initially is going to increase. But even that has long-term negative effects, because I've been watching Deadliest Catch recently. I'm obsessed. If you're out in that freezing cold temperature for too long, what happens? Hypothermia, right? Like that. Yeah. So there's something to it, the cold exposure. Again, they need to harness the correct method in inducing this cold exposure to be able to target thermogenesis, and I just don't know if it's possible. But it's there. Like the theory is there, right?
Right. It does make sense, but it's like, you're in the water for such a short amount of time that even shivering thermogenesis, I don't think is going to make a dent in metabolic rate.
Yeah.
But the high, post response of getting yourself back to baseline, ramping that metabolic rate. Again, I think the theory makes perfect sense, but the data just don't show us that. And it's like, people are willing to just, break ice with an axe and hop in this tub in the snow at 5 a.m. and just get into excruciating discomfort and then come back inside and post videos about it. But then they don't go to the gym.
Right.
It's like, if you're trying to find all these
ways
easy ways out. And that we talked about that in the podcast, but I wanted to bring that up because you can provide some layer of expertise that I wasn't able to. But
if you want to find an easy way out, you might as well just quit looking, because there's not an easy way out. There's nothing magic about being metabolically healthy. You have to put some work into it. There's just not a shortcut. It doesn't exist. And if you want to supplement your regular workouts with cold exposure, be my guest. But at the very least, it feels good.
It blunts protein synthesis and the ability to build muscle. So like, that's where it's like, okay, down the long term, here's another theory. If you're doing cold water and you're not able to build as much muscle, what's that doing to metabolic rate?
Well, and we know if it blunts your ability to synthesize protein and build muscle, we know what happens. You don't have the muscle mass that you would otherwise have. And we touched on it earlier. If your muscle mass is higher, your thermogenesis is going to increasing your whole body metabolism. That's just the way that it works. So they not a lot of forethought to that experiment. Do you know, did they do any in these studies? Are they doing any gas exchange testing?
So this particular paper, what they did is they took like swimmers in Denmark and had them go swim in the ocean, and they used cold water, they used sauna, and there was some other variable in terms of, I think, just swimming in general. And so they weren't actually studying brown adipose tissue. And that's what I was saying. Like, and as like, when you design an experiment, you have to do a power analysis directed at what you are specifically looking for. They were not looking for what happens with brown adipose tissue, like the cold water induced metabolic changes. So they found significance, but it was not powered to test that. And so like, we can't actually say that, but that's the study that blew up that everyone is obsessed about.
Got it.
I don't really recall 100% of the methodology, but
the interesting thing would be if we could find a study, or someone could do a study where they want to really dig into the thermogenic effects of cold exposure or cold water immersion. If they could figure out a way to hook somebody up to a metabolic cart and just have a resting metabolic test done at normal temperature, and then put them in the cold water and do the same thing while they're in the cold exposure, that
they've done this in exercise. So they had people exercise in cold versus exercise in room temperature. Yeah. Well, no. The swimming one. Well, I mean, like they were hooked up to a metabolic cart, and exercise in the cold actually decreased energy expenditure, and which, I mean, your max heart rate's going to go down, VO2 max, that's going to go down too in the cold. So it does make a little bit of sense, but like, metabolically, they were burning less fuel in the cold.
And the first thing that I think about with something like that is, sure, you're gonna utilize less fuel because your body doesn't need to bump up to that next level to keep you cool. You're already cold, right? So if you're exercising in the heat, your body needs to kick it into overdrive because it needs to keep you cool, well as provide you the energy to continue moving. And in the cold, you may not have to do that so much. So don't go run a marathon in 100-degree heat, but like
or freezing cold.
Yeah. So that would be my thought with cold exposure. The theory's there, but if we can, the important thing is figuring out in practice how to implement that. And as I've mentioned 100 times, is that it can't be the only thing. If it works for you, great, but it's not going to work forever, and it needs to be the supplement to the normal exercise stuff.
Yeah. I mean, and we talked in other podcasts about Lane Norton's got a really good quote of like, at that point, you're stepping over dollars to pick up pennies. Like, you're focusing on these stupid little things that it's like, we know better than any other area of research how good exercise is for you. Why are you trying to get around it? Just do the work.
And if you find what you like, then what else matters? Like, if you find an exercise that you enjoy doing, like I enjoy riding my bike. I'm on a bike because I know that it's good for me, and I know that it's doing exactly what I need it to do. Right.
And you'll keep doing it.
Right.
Because you like it. And that's what people are like, oh, do I have to go to the gym? Do I have to like do this? Like, I hate going to the gym. I hate doing high intensity. It's like, don't do that.
Yeah, right.
Just figure out how to not sit and eat, and you'll be good.
You need to eat, but you don't only sit and eat together at the same time. You need to eat, and sitting's fine, but get up once in a while. Yeah, get a standing desk. I'll build you one.
That last like 2 minutes is just like that's all we need.
What else does Reddit have to say? Nothing.
I just had those 5 questions.
All right.
Yeah. Anything we need to circle back on?
Circle back. What did I text you? Oh, this is another question I had. So tying this all back to your research, I keep coming back to this awesome new mechanism you taught me. It's funny, like getting a PhD, studying muscle. I'm taking all these physiology classes. I even called some of my colleagues to ask if they had ever heard of this, knowing that you were coming in to talk. Not a single person knew what it was. And it's just like crazy that there's this area of research that is so unknown. But anyway, that's why I'm so fascinated.
Well, part of that too, just quickly, is because, it's molecular. So it's just myosin specific, right? And usually when I talk to people about this, I start off with most muscle research only cares about active contraction. We're looking at, like I said, we do active force measurements and force generation and peak isometric force. I'm doing the complete opposite of that. So that's partly it. But also, it's pretty biophysical, right? Like using fluorescent decay measurements on a microscope is biophysics. That's what they do. So I'm really, what's happening here is I am asking physiological questions, but answering it using a biophysical tool. And I think it's not enough in either of those worlds to elicit the attention from either of those worlds. I am blending these two things together in a sense, but I just can't capture the attention of people until I start talking about it like this.
Yeah,
right.
Exactly.
So I think that's partly why people don't know about it, because those two worlds should intertwine more than they do. And I haven't seen it.
See, and I am excited to see where it goes because of how relevant it is. Right now, like, you hear about it. And like, even when I was talking to those friends, like, oh, so myosin has 2 different like states. What does that matter to me? But talking about how relevant this could potentially be to so many health issues, it's just wild. So, but another thing that is being heavily studied right now, and actually there was a lab affiliated with our lab out in Colorado that was studying this too, and that's hormone replacement in like transgender individuals. So as you transition and are supplementing with hormones to help aid from either a male to a female or female to a male. I actually don't know the findings of the lab, but when I was doing my dissertation, they had a group of rats and they were just studying like a transgender model of the rats and what happens to muscle in exercise. Again, I don't know the results, but it's just an interesting area of research that everyone's doing right now. Knowing the influence that hormones, especially estrogen, have on muscle and disordered state of myosin, like what are some potential things that are unknown with a transgender individual getting hormone replacement therapy? What are some concerns, and what are some things that maybe could help with a transition and all that?
And it's first off, I think we need to be doing way more research with transgendered individuals, especially when you're talking about an organ like muscle that takes up there's such a significant amount of something as important as metabolism, right? It is, we need more data out there. And I don't know if the stigma extends beyond just socially, if that goes into labs too, they're afraid of like ruffling feathers. But I think it's important to study, because we're only finding out more and more people are being more comfortable with being their true selves and being able to be who they actually are. And we need to understand that better. So I'll say that first. But to answer your question, I think the first thing that comes to my mind is aromatase. And aromatase is an enzyme. So the way estrogen works is it is a product of testosterone. So testosterone plus aromatase, this is probably overly simplistic, but when testosterone is broken down by aromatase, it creates estrogen. So everybody has both testosterone and estrogen. There's just different levels and effectiveness of aromatase between the two biological sexes. And I think that is an important aspect of studying transgendered individuals, because in order, especially thinking about going from transitioning from female to male, the level of aromatase, I don't know how that plays into that hormone therapy, but if their level of aromatase is elevated still after they have transitioned, their levels of estrogen are going to be elevated as well. I don't know if that's how it actually works. I don't know if there is something in the hormone therapy that counteracts that, but my head initially goes to aromatase with that because of the importance of it with as an enzyme with testosterone to make estrogen. So as far as superlax myosin goes, I think before we can really start understanding what kind of role that can play in transgendered individuals, is we need to understand estrogen and just sex hormones in general and how that affects and regulates myosin relaxed states. You know what I mean? Like we've seen it in males that are their predominant sex hormone is testosterone, and young and aged males have differing levels of testosterone, and we saw no difference between their relaxed states. But with the loss of estrogen, we see changes. So if a biologically female is transitioning to male and they're losing estrogen as a result of that because of the hormone therapy that they're on, does that also result in the destabilization of superrelaxed myosin? Where's that cutoff, right? Like that's really the question, is what is the threshold of, you know what I mean? Well, it could be a super interesting field of research to really find a threshold of at what point during the transition process does this particular tiny aspect of skeletal muscle physiology change, or does it change at all? It may not. I don't know.
See, yeah, and it's just interesting, like how quickly I think that this field is moving, a little bit ahead of the data. Yeah. And like at one hand, like you said, you want to try to provide the support to everyone that you can, but on the other hand, it's like, such taboo to talk about. Like there's some real concerns about this. You're at the front in terms of hormone influences on muscle, and there's still so much you don't know. Right. And like you saw differences between sexes, and then in certain, like even in the male mice, you had the low estrogen. And then did you give estrogen to the male? Okay. But even yet, like you've got these experiments that you still don't even know the answer. And so we're using this hormone therapy that we really don't know what's happening, especially as it relates to the muscle. Yeah. That's my concern with this. And especially then you get into a transition of an individual who's during pubescent years, and like how hormones are not just playing a role in, reproduction, but they're playing a role in
foundational development of the person. Yeah.
And yeah, so I'm curious to see how that research develops. Is anyone in your guys' lab doing experiments with that specifically, or like anyone affiliated with your guys' lab? As
far as like doing transgender research?
And just like, transgender hormones and how it influences muscle physiology.
Not transgendered research yet. I don't know if Don will take it that far, but we do some work where we're supplementing male mice with estrogen. So we'll inject estrogen treatment into male mice. That's one of our postdocs is doing that. So I don't really know. I think that's pretty early on. He's studied aromatase in those animals, and he's kind of working on some of the kind of the beginning stuff that's paving the way for future research in our lab from other people, because I think he's moving on shortly too. So I'm not really sure where his research is at, but slowly, I think our research is kind of
heading in that
direction, but only at a foundational level. I don't think that our lab will move into the transgendered physiology research sphere, just mostly because our grants are, we call it an aging grant. My PI's got 2 R01 grants. We call one of them the aging grant, and that's really where the majority of the funding comes from. So it's more of an aging thing, like the umbrella it's under is aging. So I don't think it'll end up that way, but I can certainly see how the research that we are doing now in the very near future will tie into that.
Yeah. You'll be referenced in a lot of introductions and of research papers of like, okay, this is what the theory here.
Speaking of which, the paper from Mankato has like 40 citations now.
Really? Yeah, I haven't looked at that. Look at us.
Yeah.
Look at you.
I got an H index of 2.
Oh, yeah. I don't know any more questions related to that.
So on your sheet here that you sent me, it says, what are some exciting areas of research in your field?
Yeah. Well, I would have asked that anyways.
Yeah. So I think we're kind of there, right?
Okay.
Like that.
Yeah.
We've kind of been talking about that, right? So, one thing that we didn't really touch on with this is the true effect of physical activity on relaxed myosin states. And this ties into the research that I would like to do if I had time and funds. And what I would love to do is be able to take a group of people, humans, and take a biopsy, measure the SRX in those untrained muscles, and then do just a control group and like a training study, and go 16 weeks of resistance training or high intensity interval, whatever, do a training study, and then take the control group, compare to their pre, and take the exercising group compared to their pre, and see how training truly affects
those within group.
Yeah, the within group comparisons. I can't imagine that 16 weeks would change all that much in just the control group, but
dude, I bet it will. I mean, like, you look at some of these papers that are finding.
I'm just talking like control to control 16 weeks apart. Like, I don't think that 16 weeks is not enough for just like general time to change.
Yeah. I was thinking the intervention.
But that something like that brings into, there's complexities to that, because what muscle do you use, right? If you're doing a human biopsy, you can't do soleus. You can't get at it. I don't know if you've ever seen a muscle biopsy before. I bet you can see it on YouTube. I'm not sure.
I had a bunch.
Did you have them done?
Thigh and
they were taking what, vastus lateralis, rectus femoris?
I don't know. I was a participant, and I just laid there and screamed.
Yeah.
One of my quad muscles.
So have you ever seen one, Reed? So think of those the water guns that you put the tip in the water and you go like this. And Yep. That's a muscle biopsy. That's basically
with an apple core. That's how I explained it. That like, you go in there, you and twist.
So you get a little bit of lidocaine, and they drop that needle on there and they suction it out. And it is enough to where generally you need a medical doctor present to be able to do one.
Yeah, we had a doctor.
So I would love to be able to do pre and post measurements of the SRX with exercise, with training. And actually, I mean, you could even take it the next step and have some people doing endurance training and some people doing resistance training, and seeing if there's differences there. That would be the ideal study. I was working on setting up an exercise study with our mice, doing wheel running for 12 weeks, and doing, in conjunction, parallel to that, having a group of sedentary mice where they are in a space restricted cage. So they're just their ability to move within that cage is restricted. And comparing that, but it takes so much time, and then on top of that, it's so much data to analyze, and it's sad. Yeah. I don't know. I mean, this is something kind of interesting. Just a little interesting fact: mice are incredibly athletic animals. Dude, they are insane. Female mice in any random night, any given night, so they're nocturnal. They're active at night. If you put a running wheel in their cage, over the course of their 8-hour period, or what we did, 14-hour light dark. So 14 hours light, 10 hours dark. In those 10 hours of dark, they will run a 10K.
It's wild. Those little things.
Yeah.
We did. Have you ever like done, because then that's like physical activity versus the exercise experiments where you have them on the treadmills and stuff. We used to put them on the treadmills and we'd sit there with little like broomsticks to make them, but they want to run.
They want to,
love it.
That's the nice thing about the voluntary wheels is that you don't have to worry about them like forcing them to do anything on the treadmill. Like it's pretty barbaric. I don't know if people really do it anymore, but some of those treadmills, they do forced running. And if they come to the back of the treadmill, they get zapped a little bit.
Yeah.
But the wheels, they'll just get on it and run and run and run. And then they'll get off and they're like, I want to go back on. And they'll do these different counts over, and they'll run equivalent to a 5 to 10K every single night. And they're 40 grams, man. Like, I don't think I've run a 10K total in my life. That's like, so yeah, they're pretty interesting. I just have run out of time, and on top of that, unfortunately, the microscope that I used for all of my SRX research was decommissioned not too long ago, and it had a component on there called a film detector, and a film detector is that lifetime imaging module, and there's not another one on campus. There's not another film detector, so there's not a microscope that I can use to do those experiments anymore.
It's probably good, because otherwise you would just never stop.
Right. So they replaced my microscope with a brand new fancy one, but they didn't put a film detector on it because the film detector is $130,000.
Ooh,
jeez.
So I'm kind of done collecting data, which is partly why I was excited about talking about the mitochondria stuff earlier too, was doing some mitochondrial biogenesis stuff with the tissue that I already have, because I have boxes. I probably have like 20 boxes full of muscle tissue right now that I have no plans, but would like to do something with it just to add a little bit extra to my dissertation work before I'm done.
But you have a good enough dissertation.
Do I?
I don't envy the period that you're in right now.
Yeah. So we touched on a little bit just the transgenic model too. Is that's only female mice. That I do have male mice, but I can't obviously do the muscle fiber stuff anymore. But those female mice, where we're narrowing in on the estrogen, the estrogen component of in what way is estrogen important to the super relaxed myosin? And it's not necessarily like the signaling to skeletal muscle specifically, because when I knocked out the ability to signal to muscle, the variables, those parameters of relaxed myosin stayed the same. So we're narrowing in on that, and doing more estrogen research, considering what my lab does, would be ideal too.
Are you going to do a postdoc?
Not if I can help it. I applied for a postdoc at the VA, and the VA has a group there called Recover, and it's a rehabilitation group where they're studying a million different things in veterans that have lower limb amputations. And if I got that postdoc, yeah, I'd do a postdoc, because that stuff's super cool. It's an incredible group of researchers. They have engineers, mechanical engineers, software engineers, like all these different people. And then on top of that, there's occupational therapists, physical therapists. All of them have PhDs. And I have a couple of friends who are in my program that are biomedical engineers that are working there as well. My friend Nicole did this study on developing a prosthetic foot that has a variable angle, so that women that have lower limb amputations can have this variable ankle angle to wear high heels, because prosthetic feet don't do that. So she's working on developing a way to be able to allow women again to be who they are. They want to,
that's so
even
cool.
Even if you lose your foot, you want to wear a high heel.
Yeah, we got something for you.
Yeah,
that's sweet.
So if I got that's the only postdoc I think I would take at this point, is to work there, because it's such a cool group of researchers, and it's super collaborative. And the only thing is that you're required to write grants, and we've been over this. I'm not good at it, so it's not really what I envisioned doing. But I would suffer through it is probably too strong of language, but I would do it for that job because they're just great. So no postdoc.
What's the program called?
Recover. Yep, it's a government thing, so it's an acronym for something.
When I'm at the VA, I'm gonna seek them out. So I'm sure I'll be there for a rotation.
Yep, they're super cool.
I just thought of an idea for our final question.
Did you get a new laptop?
I did. It's only like $200.
What?
It was cheap.
Is it
Kayla's like, Dad, you're done.
Is it like a what are those cheap Chromebook? Is it a Chromebook?
That's what I had. This is just a little HP, yeah.
And it's got a full Windows operating system on it?
Yeah.
Where did you get it? For $200.
Amazon. Well, $249.
You know, just a cool $50 then.
Okay, here, let me all right. So
I want to get a cheap Windows computer. I don't know if I can do it anymore, but with the woodworking stuff that I was doing, you have to some of the stuff requires Windows. They don't run on Macs, and I don't want to boot camp my Mac. So
Boot camp my Mac.
Get one of these. All right.
I think that the extra $50 might do it for me.
Yeah, I'm on 200 cap.
Okay. 1 second.
Do you want to put in your plug for your podcast?
Oh, sure. Well, Luke is pulling up whatever he's got. If you want to listen to me talk more, sorry. If you do, that's pretty incredible. I do a podcast called The Doctor and the Dog. It's me and a friend who lives out in Boston, and we mostly just talk about Minnesota sports. We've done some Viking stuff. We've done some Timberwolves stuff. We record on Monday. We're going to talk about what we got going on with the Twins right now, and a friend of ours who's a Yankees fan is going to be on the podcast. So
Yankees.
We usually, the first couple we ran about an hour because it was very unorganized, and we just like talked about whatever we wanted, but it was also an exciting time for the Vikings because it was draft time. So we rambled and we were just super excited. So we're going to keep it around 30 to 40 minutes, I think, for this next little bit until Viking season picks back up. But it's Minnesota sports, and we've only recorded a couple episodes, but eventually we're going to actually tie in things that are related to the name of the episode. It's called The Doctor and the Dog because I'm a PhD candidate. It's a little preemptive, but alliteration is important.
Yeah.
And my friend Joe, his Twitter profile picture is Joe Cool, the Snoopy alter ego. Yeah. So The Doctor and the Dog is kind of where that came from.
Oh,
so
nice.
Yeah,
where can we find you?
We're on pretty much most of the streaming platforms: Spotify, Amazon, Apple.
Hell yeah. Doctor and the Dog.
Doctor and the Dog.
That's where I'm going to get my Minnesota sports scoop skull.
So it is explicit, right? Is that the word?
Yeah.
Yeah. There's expletives.
You say real cuss words?
We say real cuss words.
Well, when you're talking Minnesota sports, it's just...
Right,
yeah, literally just like,
Do you remember me talking about seeing you at the Colts game 2 years ago? Did I tell you about that? Yeah. I don't think I ever told you. Maybe I texted. No, I texted you. I did text.
Oh, you did? Okay. That game was incredible.
It was so much fun. It was my first game at US Bank.
We were about to leave.
Same.
And we're like, let's just one more drive. And we scored and we're like, one more.
We were on the same level of seats, and I was eating a snack, and I swear to God, I'm standing at this high top table outside of my gate, or outside of my seating area or whatever. I'm eating a snack, and Reed walks by me just mad, drunk and mad.
Yeah, I was. Yeah.
You also had an edible too, right? We can take that out of the podcast, but
How'd you know that? It's legal.
Because you told me that. You said you were at the game and you left early.
You told me that too. I think you told. I think we talked about this because somebody.
Maybe he told you and you told me,
or I don't know. It doesn't matter. But just your walk by me, you're just like mad, and it was
starter jacket.
Maybe the beginning of the third quarter or something like that. So nothing had happened yet. We were there Emily bought me my ticket for my birthday, right? The game was like December 16th, 17th. My birthday is the 20th. So Emily bought me that ticket. She goes, this is your birthday present going to this game, but also your birthday present is you not having to take me. So,
That's hilarious.
so I went to that game, and funny, this probably sounds very weird to people who, to anybody, a normal person.
This probably sounds weird to everyone.
Got these tickets. I met up with people that I knew on Twitter from Vikings Twitter. That's who I went to the game with. I sat in this. I'd never met these people in person before, and they bought the tickets. I Venmoed them. They sent me the tickets, and so there's like 8 of us at this game together. That
just straight up true sports fan.
Yeah,
right
literally
there.
just like Vikings Twitter. We had talked. I mean, you talk enough to kind of really get to know people. Like, there's some people on Vikings Twitter that I will never want to meet in person, but these people, I was like, yeah, this is fine. Actually, one of the guys, his dad played for the Vikings back in the like early mid 90s, I think. So
what was it?
Jim Gustafson, who's a wide receiver. So we got to, I've hung out with his son a little bit. So Emily's texting me.
Get home. All right, this is going to be our fun thing, I think, until we figure out our fun thing. All right. So first off, thanks, Zach. This was the best.
This is great.
I have a feeling we're going to have you on once you discover that pharmaceutical drug that targets myosin, and then we can cure metabolic disease with exercise, nutrition, and a drug.
If you ever want to talk about the 3-minute all-out test, I'm all about it too.
Oh, no, man. Actually, nope. That'd be cool to do like a anaerobic power. Yeah, we'll
was
get
the worst test.
I will come back anytime. This is a blast.
You're going to be a recurrent guest too. Okay, so we're going to end with ChatGPT giving us a question. Okay. Totally random question. I just asked for a question. What is the meaning of true happiness? Can it be achieved through external circumstances, or does it require an internal state of mind?
Ooh. I didn't know that ChatGPT got so philosophical.
I said,
we're going
give me a philosophical question.
Of course you did, nerd. So read it again.
All right. What is the meaning of true happiness? Can it be achieved through external circumstances, or does it require an internal state of mind?
I think true happiness requires external circumstances and internal circumstances. You need to want to be happy in order to be happy. But you also need to allow the things around you to make you happy. And if you can't accept that, you're never going to get that. And so true happiness to me is, it's hard to define what happiness is.
Tough.
It just is. I mean, it can be achieved, but once you get there, you want more, right? It is the Matthew McConaughey thing. Who's your hero? Me 5 years from now. That's, if you're happy now, imagine how you're going to be in 5 years. Like, it could, in theory, I guess it could go either direction, but
see that's more of like the external. Like, if you're thinking in the future, it's like you need external stimuli to make you happy. I think of this as like happiness is not needing the external stimulus to be happy, right?
And
like
I think
appreciating the moment. I
think the external part is not things. It's not material things. I think it's the receiving the I'm going to use things, receiving the stuff that the world is giving you, and the people in your life, the things in your life that are making you, that are trying to make you happy. It's accepting that's what that is. Like pets or family or spouses or the Vikings. So it is not so much the physical thing, but the feeling that those things are projecting onto you.
Yeah, and that's so like, do you need those things, or can you be happy regardless of anything else in your life? Internal state.
Well, if you eliminate all of the external stimuli, what are you left with, though?
That's why this is philosophical.
Yeah, because I think if
you be happy,
I don't think you can be without external stimuli.
Now we're getting real deep,
right? Because I think about if I'm just solo, just me, there's nothing in the world like there's nothing about that could make me happy except for maybe silence. But I think there would always be something missing if all it was you. I think there's always going to be, if it's just you, there's always going to be some part of that missing. And maybe that piece is only 5% left to make you 100% happy. But other people you might need 30% more from the external stimuli. But I think there's always going to be a piece missing if you don't have something on the outside that is being pushed onto you to help you be happy. And that can be anything for anybody. And I like the first thing that I go to my wife, my future kid, and my dogs, right? Like that's like those things make me happy now, have made me happy, and maybe a better way to put it is they contribute to my happiness, because there are things that I can do without them that make me happy. They contribute to my happiness, but without having all of those together in my puzzle, my full thing, my pie chart, without having all of those together, true happiness it's not fully true happiness.
That makes perfect sense, because so the answer to the question then would be no. So what is true happiness, and can it be achieved through an internal state? And I would agree with that. And there's someone, I don't know who made this quote of like, what is happiness if you can't share it?
Yeah.
And so like people get so focused on these external things, and then you end up being like alone and you achieved all these things. And it's like, oh wait, I'm not happy. Well, so the answer is no, you do need the external.
So. Yeah. The I don't know if it's real or not, but the John Lennon deal from, the John Lennon quote.
He's got a lot of quotes, dude. He's got said a lot of things.
They say that when he was a kid at school, their teachers asked them what they wanted to be when they grew up. And, people put police officer, teacher, firefighter, whatever. John Lennon put happy. And the teacher said to him, "You don't understand the assignment." And John Lennon said, "You don't understand life," or something like that.
Wow.
So,
wow.
Imagine that, just like a little student.
Powerful.
Yeah. What if I said that to you when you were teaching me?
You don't understand life.
You're right. Man, to think about, just quickly before we end here too, just to think about, so it's 2024. We met 10 years ago.
That's a whole decade.
I was teaching biomechanics and kinesiology at Mankato when I first met you, and I met Reed probably, we might have even had, did we have class together?
We might have. I've seen you around. Yeah, but when we first initially met, I think it was on the trip, like when we started hanging out.
Leading up to the trip is when we really, because we had class before we went on the trip, right? So, but that was 10 years ago. We went through, all three of us got married. I went on 2 South Africa trips in that time. We both went to Australia. We both, the PhDs, you've been moving around. You found a job that sounds like you're in a good spot with a government job. That's incredible. Kids on the way, kids already, like a decade. In the grand scheme of things, 10 years is not that long of time, but look at the shit that has changed since the time that I was 24, like 10 years. And here we are, all back in the same room.
That's wild.
Wishing
if you would have told me 10 years ago that we'd be sitting and 3 of us having a podcast.
Wait, what's a podcast?
What's a podcast?
Well, shit, I think 2014 is roughly around the time that Serial came out, right? Like that was when the podcast world started to boom because of Sarah Koenig and doing the Serial podcast. Was that, do you guys know what I'm talking about?
No.
I heard of it. I don't really know the details.
So if you guys listen to podcasts regularly, and you like shows, especially ones that are kind of more of a story and less people bullshitting, Serial really started the movement. And Sarah Koenig talked about this kid in Baltimore who was accused of killing his girlfriend, and goes through the whole story of did he do it? Did he not do it? Why? If he did it, why did he do it? And then the court proceedings and how the how the evidence was mishandled and all this. It's an incredible podcast, and they've gone on to tell a few more stories like that. So I think that's how the podcast craze started, but it may have been earlier than 2014, but 10 years ago.
That's crazy.
Now there's so many podcasts.
Yeah.
I can't wait till the awards when it's Kickback Science and then Doctor and a Dog. We're walking down
we're up there together. Sports and health.
Yeah, the crossover. What I would like to do with the Doctor and the Dog is be able to, we have Reed's Reddit, that I would like to have some bits on mine to talk about sports science stuff, and it's just going to take a little bit of finding some of the movement data with athletes, and then I can analyze it in R, but it's just a matter of being able to get access to the data so that I can have little bits in my podcast too.
That'd be sweet. That is so unique
awesome.
too.
Yeah. So it's a work in progress, but we're getting there. So if you want to hear me,
how often are you going to release the episodes? I guess it probably depends on the season.
It's pretty sporadic. Yeah. I think it'll be pretty regular during the Viking season, because there's so much to maybe like on Monday, or the day after the game we'll release. But right now we're kind of heading towards the sports abyss where not a lot of stuff is happening other than
just
the
baseball,
Twins.
yeah.
And I'm a huge baseball guy, but it's a long season. It can be pretty mundane. So I think for the rest of the summer, we're going to talk to friends of ours that are sports fans and just
get people riled up about sports.
Yeah, like I said, we have a Yankees fan coming on Monday, so that'll be pretty fun.
Fuck the Yankees.
Yeah.
I love it.
Well, thanks again, guys.
Yeah,
I appreciate
you coming on.
Appreciate the time. I took a lot of your time, but hopefully I
great.
taught someone
we
something.
We took a lot of your time.
Yeah,
you didn't take our time.
In summary, in conclusion, exercise, eat good food, don't try to get the quick way out, and supplements are a crock of shit.
You heard it here first from Zach, and we'll hear from him again. All right, thanks for tuning in to another Kickback Science episode. I'll miss you guys.
Miss you then. See you.
Thanks again for tuning in to the Kickback Science Podcast. We love talking science with all of you, and if you feel the same, you can support us by subscribing, hitting that like button, and giving us a follow on our social media pages. Don't forget, you can submit questions through our website and in the comments section of all our social media. Have a good one, everyone.